Okay, so welcome everyone to the 55th lecture of Dr. Hydri Step 1. Before we begin, can we assure that you guys can hear my voice? Can you guys hear my voice? Is my voice clear? Okay, good, very good. How are you guys? I hope you guys had a long, good weekend. Um, we have 8 students over here. We're gonna wait for 2 more minutes for the rest of the students to come. After that, we will join. Is my voice breaking? Is everything okay with my voice so that we can begin today's lecture? Is my voice breaking? Do I have a broken voice? Okay, perfect. Okay. Okay, so before we begin, I just want to make sure if you guys did the UWorld questions yesterday, throughout the weekend. If you guys did, then did you guys do questions from the biochemistry genetics? And how many questions did you guys do? And how was your score in the blocks in which you did the questions? Everyone else, how many questions did you guys do during this whole week? 10 questions. Okay, not bad. Anyone else? How many questions did you guys do? Offline questions. Okay. 40 questions. Okay, that is very good. Out of those 40 questions, were they all from genetics or where were they from? Okay, mixed. No problem. Okay, so good, very good. So we are going to start with the laboratory techniques from biochemistry today. And it's a pretty difficult topic, but we're going to try to break it down into easy, digestible form so that everyone can understand and associate all the questions with the topic. So before we do that, can we do a quick revision and recapitulation of the previous lecture to see if we all are on the same page, yes or no? Can we do a quick revision and recapitulation of the previous lectures? Is everyone ready? Okay, good. Okay. So you have a patient who comes to your clinic. The patient has complaints of infertility. The patient has the apex beat in the right fifth intercostal space, and the patient has long-term chronic lung infections. What is your most probable diagnosis? Okay, good, very good. You have a patient who comes to your clinic with chronic lung infections and infertility, and along with that, the patient has pancreatic insufficiency. What is your most probable diagnosis? Okay, good. Okay, you have a patient who comes to your clinic with blue sclera, multiple fractures, poor dental, poor dentures. Okay. Okay, good. You have a patient who comes to your clinic. The patient has murmur in the heart, along with the fact that the patient has hypermobile joints, history of dislocations, and very good. Now you have another patient in your clinic who is 99th percentile for his height. The patient has pectus carinatum and pectus excavatum. Along with that, the patient has murmurs. Very good. Marfan syndrome. Very good. You have a patient who comes to your clinic um, with breath sounds of vesicular with prolonged expiration. Along with that, the chest X-ray shows hyper shows that the lung findings are the fact that they are um, hyperdense. There is tubular heart, and the patient also has The patient also has liver failure. The patient also has liver failure. What is your most probable diagnosis? Liver failure with lung problems. Liver failure with lung problems. Anyone? Do I have anyone with any probable diagnosis for this one? Liver failure with lung problems. Alpha-1 antitrypsin deficiency. Very good. Alpha-1 antitrypsin deficiency. Okay, good. Very good. Next one is you have a patient. You have a patient, and in that patient, you have lacking of DNA replicating enzyme, which is responsible for unwinding of the DNA. You have a deficiency of an enzyme, which is responsible for unwinding of the DNA. Good. Bloom syndrome. Very good. Okay. You have a patient who comes to your clinic with subluxation of the lens, and the lens subluxation is upwards and outwards. What is your diagnosis? Upwards and outwards. How about the lens dislocation is inwards and medially? Okay, good, very good. Okay. What is the genetic mutation for Marfan syndrome? What is the genetic mutation for Marfan syndrome? Which gene is affected? FBN1 gene for fibrillin scaffolding protein. Okay, good, very good. What is the genetic mutation for osteogenesis imperfecta? Okay, what is the genetic mutation for Ehlers-Danlos syndrome, the classical type? Okay. What is the genetic mutation for the vascular type of Ehlers-Danlos syndrome? Okay, good. You have a patient who comes to your clinic with multiple bruising, impaired wound healing, gum bleeding. At which stage of collagen synthesis is most primarily the affected process is? Very good. Hydroxylation. Okay, good. Good. Hydroxylation. The students who are not present at the previous lectures, did you get a chance to go through our lecture videos, or are you guys did you guys go through first aid by yourself? Did you guys go through first aid by yourself, or did you guys go through our lecture videos? For the students who are not there in the previous lectures. Okay. Having said that, I just want to make sure when you went through the video, did you guys have any problems when you went through the video, or did you guys understand the video and the lectures, and did it help you read first aid properly? Did it help you read first aid properly? Okay, good. That's very good to know. Thank you so much. Next one. Next one is you have a patient who comes to your clinic with you have a patient who comes to your clinic with multiple lesions of his skin, which are there when he is exposed to the sun. At which stage of DNA repair is the problem? At which stage of DNA repair, or which stage of DNA repair is not working? Okay, good. You have a patient who comes to your clinic with a family history of colorectal carcinoma, and at which stage of DNA repair is the problem in? Family history of colorectal carcinoma in 3 generations of 2 affected individuals, and 1 patient is above less than the age of 50 years. Very good. Mismatch repair. Mismatch repair. Okay, good. Very good. Next one is you have a patient who comes to your clinic with ataxia and then spider angiomatosis. The patient has skin reactions when the patient goes through X-rays. At which stage? Good. Very good. Okay. Okay. Okay, non-homologous and joining repair. Okay, good. Okay, so with that being said, are you guys well aware of the types of questions which you will receive from genetics? Okay. Okay, almost. So basically, these are the types of questions, more or less, which you will be received from genetics, which will come in multiple patterns. But if you have understood the lecture and if you have understood all the processes, and if you can review genetics once again after we are done, then hopefully you will not have any problems with any questions. Okay. Can you repeat the symptoms of alpha-1 antitrypsin? We will repeat the symptoms of alpha-1 antitrypsin once again when we reach alpha-1 antitrypsin. But the sign symptoms are basically you have sign symptoms of COPD and you have sign symptoms of liver failure. Okay, you have sign symptoms of liver failure. So if you have a patient who have sign symptoms of those two together, then high possibility it's alpha-1 antitrypsin. Next one. I didn't get a question about Bloom syndrome. Bloom syndrome, you will receive questions about Bloom syndrome in MBOS, not in UWorld. Okay, in UWorld, the question will come according to the absence of the DNA enzyme. And if they ask you which DNA enzyme is absent, which is responsible for unwinding, then the DNA is the DNA helicase and the syndrome is Bloom syndrome. That's that. Okay. Okay. So with that being said, it's 9:15 as of right now. We want to get ahead on our schedule on first aid. So without any further ado, do we begin today's lecture? Yes or no? Can we begin today's lecture? Okay, good. So let's begin with today's lecture. Let me share my screen 1 second. Okay. Okay. So can you guys see my screen over here? Yes or no? Can you guys see my screen? Can you guys see my screen over here? Yes. Okay, good. So what we are going to start is biochemistry laboratory techniques. Okay, I know that it's a bit difficult of a topic, but let me tell you the easiest way to learn this so that it will be very easy for you to review this part of the first aid very quickly when you do another session of, or when you do another read of the first aid. Okay, so without further ado, what I would need you guys to do is I would need you guys to have a blank page over here. Okay, I would need you guys to have a blank page over here because the only questions you will receive from laboratory techniques is you have to associate the names. You have to associate the names with the condition that's happening. So basically, you need to understand which process is responsible for which function. Which process is responsible for which function. So basically, if you are talking about polymerase chain reaction, the function of polymerase is at DNA amplification. So over here, what we're going to do is we're going to have our own notes so that it's easy for you to do a fast review. So you will have name of procedure, and function. What is the function? So, okay, so that's what you are going to do. And with that being said, are you guys ready with your notes, with your notebook, your note copy? Yes or no? Okay. Okay, very well. Then let's begin. The first thing there, the first technique that we're going to talk about today is polymerase chain reaction. Polymerase chain reaction is basically a DNA amplification procedure, right? So what happens over here is that it's a molecular biological lab procedure which is used to amplify a desired fragment of a DNA. For example, in forensic medicine, let's say you are a forensic individual and you go to a crime site, right? You go to a crime site. For example, you see that there's a dead body, right? And over here, you have, let's say, a gun, right? And in that gun, you have a tiny speck of blood, right? Now you want to make sure if this blood is the patient's blood or your victim's blood or the blood of the person or who is responsible for the homicide. So what you do is you take a small part of the DNA of this one, right? And then you amplify the DNA, right? And then you also do the same thing for this patient. You take a small part and then you amplify the DNA and then you match whether the two DNAs are the same or not. So that this is one use of the PCR. And another use is it is in clinical medicine, it is used as a diagnostic tool. For example, neonatal HIV or herpes encephalitis. So what happens is basically you take a small fragment of the DNA and then you try to amplify that DNA, right? So DNA amplification. After you try to amplify the DNA, you enter this amplified DNA in an electrophoresis. When you do that, when you enter it into an electrophoresis, then over there you can see whether the fragmented portion is amplified or not. And this amplification can help you identify what type of problem this patient is going through. Okay, I know it sounds a bit confusing, but it will sound a lot better after we are done. Okay, so basically this is used as a diagnostic tool for neonatal HIV and herpes encephalitis. The steps are basically that you have three steps. At first you have denaturation, then annealing. After that you have elongation. So for denaturation, at first, the first process is the DNA will be heated to 95 degrees Celsius to separate the strands. So you have to separate the two DNA strands. So this is a double-stranded DNA, which is heated to 95 degrees. After you heat it to 95 degrees, it's separated to two strands. After it's separated, the second step is addition of a DNA primer. And we know that primers are basically added so that the DNA polymerase can form the new structures, right? So the sample is cooled to 55 degrees Celsius because this is the temperature at which the DNA primer should be added, okay? And then after that, you need a DNA polymerase enzyme and a deoxynucleotide triphosphates. Basically, deoxynucleotide triphosphates are added to form the bonds between the subsequent nucleotides. And then the name for the DNA polymerase, which is used for PCR, is known as TAQ. This is not very high yield, but it's just for your understanding that. So after that, the DNA primers will anneal to the specific sequence to be amplified on each strand. They will be amplified on each strand. Next one is elongation. Elongation is after you cool it to 55 degrees, after you cool the primers to 55, then you increase the temperature to 72 degrees Celsius. This increase in temperature will allow the DNA polymerase to attach the deoxyribonucleotide triphosphate to the strands to replicate the sequence after each primer. So what happens is, step by step, number one, you heat the DNA to 95 degrees, which separates it into two strands. After you heat it to 95 degrees, then what happens is you cool the DNA to 55 so that primers can be added. After you add the primers, you add the polymerase and the DNTPs, and then you increase the temperature to 72 degrees at which DNA elongation can occur. Okay, and this heating and cooling will continue until the DNA sample size is sufficient to be acknowledged. Sufficient to be acknowledged. Okay, so basically what happens over here is, if we have to name the procedure, the first thing that we learned is PCR. Okay, what is the function? The function of PCR, it's used as a diagnostic tool. It's used as a diagnostic tool. And DNA amplification. DNA amplification. Okay, now the thing about the thing is, if we want to dig into a little bit deeper. So first of all, the only questions you will get from over here is basically UWorld will ask you questions that what is the procedure that we have used for diagnosis, or what is the procedure that we have used for DNA amplification, and your answer is PCR. Okay. And if it tries to go any deeper, then we can try to identify this. For example, let's say that you have a double-stranded DNA. Let's talk about the steps in the blank page. Okay, so this is the double-stranded DNA. You heat it to 95, and then you separate the two strands. Then you cool it to 55, and at 55, you had the additions of the primers and the polymerase and the DNTPs. And then you increase it to 72, at which you have a new strand. You have two new strands, okay? You have two new strands. So these are the two new strands which are amplified from this one over here. After this, if you enter this strand into an electrophoresis, right, over there, if you see that in one of those strands, you have amplifications, for example, you have, let's say this is chromosome number 1, 2, 3, 4, 5, 6, right? And then so far and so forth. Let's say, for example, at chromosome number 21, this is chromosome number 21, okay? This is chromosome number 21. At chromosome number 21, you have amplifications of DNA. You have DNA amplifications like this. So can this procedure be used to diagnose that this patient has excessive DNA at chromosome number 21? Yes or no? For example, this is just an example. Yes. So can this be used to identify that this patient is your patient of trisomy 21, that is Down syndrome? Okay. Okay. Once again, after you identify or after you amplify the DNA, then over here at chromosome number 4, if you have amplification, of DNA, right, and everything else is normal, and this one is larger than its size, then is there a possibility that we can say that at chromosome number 4 you have extra DNA? Yes or no? You have extra DNA at chromosome number 4 if this portion is identified? Yes. So do you guys remember that in Huntington's disease, we had trinucleotides repeats of CAG at chromosome number 4, trinucleotides repeat of CAG, like this one, CAG, and then CAG, trinucleotides repeat? Yes. So if you have a patient, for example, in amniocentesis, you do an amniocentesis, and then you take a small portion of the baby's DNA, and then you amplify the DNA. And over here, you see that a small portion is amplified at chromosome number 4. Then you can identify that there's a possibility that this baby has a possibility of developing Huntington's because they have a trinucleotide repeat disorder at chromosome number 4. So that is the way, this is one of the ways you can use polymerase chain reaction in a clinical setting. Are we clear about PCR? Okay. Are we clear about PCR? So what are the enzymes, or what do we need for a PCR to take place? What do we need for PCRs to take place? Number one, we need a double-stranded DNA. Number one, we need a double-stranded DNA. Number two, we need DNA polymerase. Number three, we need DTNPs. This is also another question from UWorld, that these are the three things that you need in order for PCR to take place. They might ask you that out of which of these answer options, A, B, C, D, E, is responsible for DNA polymerase to take place. And over there, you can have either one of them, and then either one of them could be your correct answer. So you need a double-stranded DNA polymerase, which is also known as TAQ for PCR, and DTNPs. Are we clear about this? DTNPs are required to form the phosphodiesterase bond in between the nucleotides. Okay, in between the nucleotides. Primer. Yes, we also need a primer over here. But these are the most frequently asked ones, these three. You can also add a primer over here, no problem. but these are the most frequently asked, okay? DNA primers are also needed, okay? But these three are more important, okay? Double-stranded DNA, DNA polymerase, and DTMPs. Okay. Okay, good. Next one. Next one that we want to talk about is RT-PCR. RT-PCR, or reverse transcriptase polymerase chain reaction. The only question you will get over here for RT-PCR is, okay, put your attention to this one. They will have a lot of information in the question, okay? They will tell you in UWorld a lot of things over here, that there's a researcher, he's doing this, he's doing that, and they will try to make the question sound as complex and complicated as possible. But at the last portion of the question, they will ask you, what is the method of detection of the mRNA? What is the method of detection of the mRNA? Whenever you see a question where they are asking you to detect mRNA, the first lab technique that should come to your mind is RT-PCR. Okay, so just write it down over here real quick before we go to first aid, is that RT-PCR is used for detection of mRNA. Okay, basically RT-PCR is exactly the same process as PCR, but it's a reverse process. What happens is over here in RT-PCR, you detect and quantify the mRNA levels in a sample. It uses a reverse transcription to create a complementary DNA template that is amplified via the standard PCR procedures. So over here, what you do is, in order to what you do is you take a DNA strand, right? You take a DNA strand, and from the DNA strand, instead of producing this one, right, you produce mRNA by using reverse transcriptase. With the use of reverse transcriptase, so transcription is basically mRNA to protein, right? And then over here, you use the reverse transcriptase to create a complementary DNA template. Over there, you detect the amount of mRNA levels. Okay, so reverse transcription is the opposite of transcription. Okay, transcription is basically you have mRNA from DNA. Right over here, you have the DNA from the mRNA. It's a reverse procedure. So, for example, the best way that you can use this in a clinical setting, okay, the best way that you can use this in a clinical setting is, for example, when you are, let's say you are an oncologist. Okay, let's say that you are an oncologist, and right now you want to see whether the oncological drug or the chemotherapy which you are using, if it is working in the patient or not. Okay, if it is working in the patient or not. So basically, what you want to do is you want to detect the mRNA level, because if you have mRNA, and that mRNA will result in the formation of the protein, right? And for example, this mRNA, let's say that you are using a chemotherapy drug for a liver cancer. Okay, for a liver cancer, you're using a chemotherapy drug. And if there's a liver cancer in this patient, then the mRNA should be extremely high. So the protein is actually a carcinoma protein or carcinogenic protein. Okay, so this should also be very high, right? Right now, if you want to see if your drug is working or not, so let's say you are using a drug, which is drug A, okay? And right now you want to detect whether the drug A is working properly or not. So what you do is you take a small piece of that tumor, okay? You take a small piece of that tumor, then you take the tumor DNA, and from the tumor DNA, you use RT-PCR to identify the levels of mRNA. You identify the level of mRNA. So let's say that the patient previously 6 months ago, okay, this is 6 months ago before his treatment started, he had 10 mRNA. And right now, after 6 months, from the tumor, you take the mRNA, and right now the patient has 3 mRNA. Okay, then this will help you identify that the drug A is working and the patient is responsive to the chemotherapy. This is one of the reasons, and this is one of the way in a clinical setting you can use RT-PCR to identify whether the, for example, that whether a carcinoma is responsive to a chemotherapy treatment or not. So you can identify the levels of mRNA. Are we clear about this? Yes or no? Are we clear about this? Okay. In order to detect the new strains of the coronavirus, okay, I'm not sure if you guys will receive questions about the coronavirus in your exams, but if you guys do, then I want you guys to know that the confirmatory process to detect the coronavirus level is also via RT-PCR. Via RT-PCR. So what we do is we take a small amount of the patient's DNA sample, right? For example, we can take the patient's sputum or saliva or nasal swab, and we take the DNA from there. We try to detect if there is any genetic finding of the coronavirus. For example, you take a part of the nasal swab, the sputum, or anything else, and from there you take you find the nucleic acids of the coronavirus, and from there, if you find it, then the patient is positive for coronavirus. Okay, but what RT-PCR will let you identify is RT-PCR will let you identify the viral load, that how much of the coronavirus is actually there in this patient. So what you do is you do the RT-PCR. From there, you find the levels of the mRNA of the coronavirus. If it's a high amount of mRNA of coronavirus, then this patient has high viral load and he is highly infective. And this is also the same way that you can use RT-PCR in the clinical settings of coronavirus to come up with vaccines and everything, because the vaccines will go and target the viruses. And in order for you to see if the vaccines are working or not, you can do an RT-PCR to see the levels of the mRNA. If there's a high level of mRNA, then the vaccines are not working. If the levels of mRNA goes down in a patient with previous coronavirus, then the vaccines are working. So this is the way you can use the lab techniques, RT-PCR, in a clinical setting to identify multiple types of treatment responses. Okay, are we clear about this? Yes or no? Yes. But the only question you will get from UWorld, is basically the name RT-PCR, and they will ask you which procedure is used for detection of mRNA, and the name of the procedure is RT-PCR. Okay, so that's that. Okay, next one. Next one is a very complicated procedure, but this is beyond the scope of step one. Okay, this is a very complicated procedure, but it's beyond the scope of step one. But all we have to identify from this one is the function of this procedure. This procedure is known as Crispr Cas9. Crispr Cas9 is basically a modern genome editing tool, which is derived from a bacteria. Okay, so basically this consists of a guided RNA, which is known as gRNA. So this is a guided RNA, okay? And this guided RNA is complementary to a target DNA sequence. This is complementary to a target. So for example, this is the part of the DNA which you are targeting. So this gRNA, or a guided RNA, is complementary to this DNA sequence. And an endonuclease, so basically Cas9 is the endonuclease, which is responsible for breaking down. And an endonuclease which makes a single or double-stranded break at the target site. So for example, after the gRNA attaches to the complementary DNA, the endonuclease will form a break. It will break it down at the target site. After you break down, the breaks imperfectly are repaired by non-homologous end joining repair, okay, which is a double-stranded DNA repair, and accidental frame shift mutations, knockouts, For example, after you break it down, then there is an NHEJ, and then there is accidental frame shift mutations or donor DNA sequence that can be added to fill the gap using the homology directed repair. Okay, so what happens is, let's say, let me break it down for you. Okay, for example, in vitro fertilization. Okay, what happened was before in vitro fertilization, you had an ovum, and then you had a sperm, right? And then via out of the mother's body in the lab, for example, this is the ovum, okay, and this is the needle. This is the needle which contains the sperm, okay, which contains the sperm. And this is in vitro fertilization, okay? So what happened was, after, I mean, before CRISPR Cas9 was there, this was normally a normal procedure, right? That the egg will be fertilized by the sperm in a lab setting, right? But after this editing software was created, the sperm entered the ovum. But after it did, this product went through the process of CRISPR-Cas9 for genomic modification, for gene modification. For example, let's say that in a lab procedure, you have a mother, okay? You have, let's say, Jane, okay? Jane goes to the lab. Now, Jane wants to get pregnant, but Jane does not want to get married. So Jane goes to the lab, gives her ovum to the lab people, okay? And they enter the sperm from any sperm bank, but Jane tells the lab people that I want a baby with red hair. I want a baby with red hair. And Jane pays them a lot of money, okay? So what happens is, for example, Jane does not have any red hair, nor the sperm over here, it does not have DNA for red hair either. So what happens is, when the lab people, when they enter the sperm into Jane's ovum, after entering the sperm, they make the product go through the process of CRISPR-Cas9, where they add the guided RNA, then they break it at the strands by the enzyme DNA endonuclease, and then they add this donor DNA, which contains the red hair. So by the help of this editing software or this editing tool, they add the red hair DNA, and then this zygote now has a red hair or a red hair DNA. So when the zygote gets older and it becomes a full-blown child, there's a high possibility that this child will now have red hair, although the parents, I mean Jane, or neither the guy who was responsible for donation of the sperm, they did not have red hair. This is just an example, and this is how CRISPR-Cas9 is used to modify the genes. Are we clear about this? Are we clear about this? Yes or no? CRISPR-Cas9. This is very complicated, but it's the only thing over here is CRISPR-Cas9. Okay, just write it down over here. CRISPR. I will repeat this, no problem. Just write it down first. Write this down. CRISPR-Cas9. I will repeat it, no problem. I know it's a bit complicated. I will repeat it. Just write it down over here. CRISPR-Cas9. The use of CRISPR-Cas9 is remove virulence factors, removing virulence factors from pathogens. Then over here is replace diseased alleles with healthy alleles. replace diseased alleles with healthy alleles. And the next one is target tumor cells. Targeting tumor cells. Removing of virulence factors from pathogens, replacing diseased allele with healthy alleles, and targeting tumor cells. Okay. Once again, let me repeat Crispr Cas9. Okay. Before I do that, okay, let me ask you another question. Do you guys have Netflix? Do you guys have Netflix? Netflix. Does anyone do Netflix over here? Okay. Okay. If you guys are actively watching Netflix, then there's an entire episode of Crispr Cas9 on Unexplained. There's this series in Netflix called Unexplained. The first episode is based on Crispr Cas9. Have you guys seen that episode? Yes or no? I doubt it. Have you guys seen that episode? Okay, good. If you guys get the time after this lecture today, if you guys want to go and chill, okay, just watch that episode of Netflix, and over there you can see that they talk about Crispr Cas9. Once again, basically what Crispr Cas9 is it's a genome editing tool. It's a genome editing tool. So basically, if you want to come up with a new protein, right? So you know, like how we have DNA, and that DNA is responsible for the protein, right? Yes or no? You have a DNA, and that DNA will form a protein. Yes or no? Can I get some active participations, please? Yes. Okay. If you change a portion of this DNA, for example, DNA A, okay, DNA A is responsible for protein A. If you change this DNA right now, if you change a nucleotide, right, over here, let's say that you have a nucleotide, right? Let's say GAA. And if you use CRISPR Cas9 to change this nucleotide sequence, right? And right now over here in this portion, you have, let's say A, for example, then the whole DNA is changed. Basically, the whole DNA is right now DNA B, and the protein is protein B. Okay, the only way that you can induce this type of changes in a DNA is by the use of the software, the use of this tool, that is CRISPR Cas9. So basically the steps of CRISPR Cas9, the steps of CRISPR Cas9 is, can you guys just give me 1 second, please? Just give me 1 second. Can you guys hear my voice now? Yes or no? Okay, good. My apologies. Okay, good. So the process of CRISPR Cas9 for everyone who wanted me to repeat the process, okay? So step by step, basically you have a complementary DNA, I mean, you have a patient's DNA. From the patient's DNA, you add a guided RNA to form another strand, okay? After you form another strand, you use DNA endonuclease to break it down into two fragments. One fragment undergoes NHEJ, which is non-homologous end joining. Another one goes through DNA addition. This DNA that is added, this DNA is added in order to modify the protein. This DNA is added in order to modify the protein. So what happens is over here, when you add the DNA, then the entire nucleotide sequence is changed. So for example, before the nucleotide sequence was before the nucleotide sequence was read in a certain manner, but now you have another piece of nucleotide added, and then over there you have this another piece of nucleotide that is added, and now the whole protein is changed. Basically, it's now a new protein entirely by itself. Okay, so you can use this procedure to remove the virulence factors from the pathogens. For example, in the DNA of a one pathogen, you have this type of DNA in the pathogen, which is the virulence factor. So by the help of the knockout mutation, it goes through two procedures, NHEJ and HDR. Over here in one, by the help of this process, you can get the virulence factor removed from the pathogen, or by the process of addition that is knocked in, you can add a healthy allele instead of a diseased allele. So the diseased allele will get knocked out by NHEJ, and then after that, you can add a healthy allele. And by this way, what you can do is you can remove the virulence factors from the pathogens. You can replace the diseased allele with healthy alleles, and you can also target tumor cells. How can you target tumor cells? Let's say that in a patient you have a genetic tendency of developing a carcinoma, okay? What you can do is, with the help of CRISPR Cas9, you can remove that DNA that will be responsible for giving rise to the carcinoma, and you can remove that carcinomic DNA with a healthy DNA. That's how you can use CRISPR Cas9. But having said that, the only questions you will receive from URL from CRISPR Cas9 is at least one, or at maximum, you will receive two questions. And both of those questions are identification. That is, the answer is CRISPR Cas9, and the question is, what procedure will you use in order to remove the virulence factor from pathogen? That is one question. Another question you will get is, what is the name of the procedure that is used for replacement of diseased alleles with healthy alleles? And this is also CRISPR Cas9. So basically, the answer is the name of the procedure, and the questions are the functions. The questions are from over here, and the answer is CRISPR Cas9. Although having said that, this is an extremely rare procedure, and you will not be tested on this. Highly likely you will not receive one question from CRISPR Cas9 in your actual exam, because it's a very complicated procedure. Are we clear about this? But the only things you have to know from CRISPR Cas9 is the function. The function is removing virulence factor, replacement of diseased allele with healthy alleles, and targeting the tumor cells. Okay? Are we clear about this? Yes or no? Are we clear about this? Okay. Okay. Next one. Next one is blotting procedures. Blotting procedures, extremely high yield. Okay, this is very, very high yield. Blotting procedure. Blotting procedure is basically what you do is, this is actually very, very simple. The simplest part is over here. This procedure is used for all the 4 types of blotting. Okay, so before we talk about the blotting procedures, I want to talk about one. One mnemonic. Okay, one mnemonic over here. The mnemonic that I want to talk about is snow, drop. Okay, snowdrop. Snowdrop. Okay, the reason why I'm talking about snowdrop is S is for southern blot. S will detect D, meaning DNA. Southern blot will detect DNA. Next one is N. N is for northern blot. Northern blot will detect RNA. Okay. Then you have western blot. Western blot will detect protein. Western blot will detect protein. Okay. So, and then you have another one, which is southwestern blot. Southwestern blot will detect DNA binding protein. Southwestern blot will detect DNA binding protein. So basically, the procedure of blotting. Blotting is basically you have a DNA. The DNA is enzymatically cleaved into smaller pieces. So basically, you break down the DNA into small pieces, and then they are separated on a gel by electrophoresis, and they are transferred to a filter. So you have any segment of a DNA, and then you separate them, and then you transport them to a filter. After you transport it to a filter, the DNA is exposed to radio-level DNA probe that will recognize and anneal to its complementary strand. So you have a radio-level DNA probe, right? For example, you have you have a DNA probe that is responsible for identification. This is your sample DNA. Okay, this is your sample DNA, and you have a DNA probe with a radio label. Let's say you have T, C, uh, TC, and then you have G, for example. Okay, so you have TCG. So you want to add TCG over here. So at first, this is broken down. Enzymatically, it's cleaved, and then you have these two segments of the DNA. Over here, this TCG, this is going to go and it's going to go and find its complementary strand. So over here, if you have any DNA with the nucleotide sequence of, let's say, A, G, C, this TCG can go and now bind to this one over here because it's complementary. T, C, G. So this TCG, this radio label TCG, is used to identify the nucleotide sequence that is AGC, because ATCG, basically thymine, will always go and bind to adenine, C will always go and bind to G, G will always go and bind to C. So you are using this radio label DNA to identify the nucleotide sequence. For example, you want to identify this nucleotide sequence, so what you do is this will result in a double-stranded labeled piece of DNA, which is visualized when the filter is exposed to the film. Now, this is identified, this nucleotide sequence is identified when the filter is exposed to a film. So by this way, you can use this process to identify this type of nucleotide sequences. If you want to identify the DNA, the blotting which you will use is southern blot. In the same way, if you want to use a sample of RNA, then the blot that you will use is northern blot. Once again, in the same way, if you want to use protein instead of DNA or RNA, the blot that you will use blot is western blot. Okay, for example, you now use western blot. Drops of the western blot in a protein sample. Okay, so this is the protein sample. You put the western blot over here, then you separate the strands, you add the nucleotide sequence, and then you can identify the protein. So this is used to identify the protein. Next one is you can also use southwestern blot to identify the DNA binding proteins. Over here, the names of the DNA binding proteins is extremely high yield, very, very, very high yield. The proteins are basically C-jun, CJU, and C-jun, and C-phos. C-phos and C-jun. So I need you guys to remember that C-jun and C-phos, these are DNA binding proteins. There's a very high yield question in UWorld where you will get a very complicated question about a research assistant who is doing a procedure. And over there, they are going to, they want you to identify that which type of blot will you use to identify C-jun and C-phos. Whenever they mention C-jun and C-phos in the questions and they ask you for the blot, first of all, I want you guys to remember that C-jun and C-phos, they are DNA binding proteins. C-Jun and C-Phos, they are DNA binding proteins, and the blot that you will use is southwestern blot. Southwestern blot, that's that. And if they want to identify which type of RNA is produced or mRNA is produced, the blot is northern blot. If they want to identify which type of protein is produced and they want to use a blot, the blot is western blot. And the same way, if they want to identify a DNA, then the blot is southern blot. Okay? Are we clear about this? Yes or no? Are we clear about this? Please unmute. Please mute yourselves if you have any background noises. I insist. Okay, please, I insist you to mute yourself if any background noises. Okay. Has everyone understood the blotting procedures? Has everyone understood the blotting procedures? Okay. So the only thing that you have to remember from the blotting procedure is snowdrop. Okay, snowdrop. Snowdrop is which type of blot is used to identify which type of structures. And if you can remember this, then that's all you need to know. That is southern for DNA, northern for RNA, western for protein, and southwestern for DNA binding proteins. Out of those DNA binding proteins, which type of DNA binding proteins will you receive the questions from in UWorld? Fast answers, please. Which two DNA binding proteins are important for UWorld? Which two DNA binding proteins are important for UWorld? C-Jun and C-Phos. Very good. Okay, good. So before we move on forward, can I give you guys 2 minutes to read this out? Okay, because I don't want you guys to get overwhelmed with the functions. Okay, I'm going to give you guys 2 minutes to read the functions, because the functions are the only thing you will receive your questions in UWorld from. The questions will come from this side, and the answers are the names of the procedure. Okay, so if you want to test yourself, block this one with your hand, use this part of the notes to see whether you can remember the functions or not. Okay, I'll give you guys exactly 1 minute, 2 minutes. I'll give you guys 2 minutes. Okay. Okay. Is everyone ready? Is everyone ready? Yes. What is the function of PCR, please? What is the function of PCR? What is the function of PCR? Diagnostic tool and DNA amplification. Very good. What is the function of RT-PCR? What is the function of RT-PCR? Very good. Detection of mRNA. What is the function of Crispr Cas9? Crispr Cas9. Very good. What is the function of southern blot? Southern blotting procedures. What is the function of southwestern blotting procedures? What is the function of northern blotting procedure? What is the function of western blotting procedure? Okay. If you want to identify C-Jun and C-Fos, which type of blotting procedure will you use? Southwestern. Okay. Southwestern blot. Very good. Okay, next one. Next one is flow cytometry. Flow cytometry is very easy to understand. Okay, flow cytometry is basically, this is a laboratory, this is a lab procedure which is used to assess the size, granularity, and protein expression. So you identify the size, granularity, and protein expression of individual cells. So what you do is, you have a cell over here. You have a cells. The cells are targeted with antibodies. So for example, this cell over here, okay? This cell has a certain antigen. So you want to target the antigen to see whether the antigen is present or not. In order to identify the antigen, you add an antibody. And that antibody is no regular antibody. That antibody is labeled with a fluorescence. So the antibodies are tagged with the fluorescent antigens, and the sample is analyzed over here, right? So you add the antigen and the antibody, and if there is an antigen in the cell, the antibodies will go and attack the antigens, and they will form an antigen-antibody complex. And when they will go down through over here, the laser, there will be a laser which is used to detect this fluorescent label. If they can detect the binding of the fluorescent labeled antibody to the antigen with the help of a detector, then this will help form a graph like this. This will help from a graph like this. So what happens is the cells are tagged with antibodies specific to the target intracellular proteins. The antibodies are then tagged with a unique fluorescent dye. The sample is analyzed one cell at a time by focusing a laser on the cell and measuring the light scatter and intensity of fluorescence. So if there is a very good binding of the antigen and the antibody, then this will result in high intensity. If there is a very minimum binding of the antigen and the antibody, this will result in a low intensity fluorescence. After the detection, the data, they're plotted either as a histogram or as a scatter plot. So what happens is over here, you will receive multiple questions from flow cytometry based on these ones over here, okay? So what you have is, for example, over here, let's say that this is a histogram, okay? The first and foremost, the flow cytometry, they are used to diagnose hematological abnormalities. So for example, in patients of paroxysmal nocturnal hemoglobinuria, you have certain clustered differentiated cells, which can be detected, right? Then leukemias, there are certain types of antigens which are bound to the RBCs which you want to detect. So what happens is over here, let's say that over here you have CD3, over here you have CD8, right? And this is CD3 positive, this is CD8 positive. And you have 4 segments of the graph, okay? If it's only over here, okay, then this is one type of finding. If this is only over here, then this is another type of finding. Then over here, if it's over here, then it's one type of finding. And if it's over here, then it's another type of finding. So each and every type of segments will result in different types of finding, meaning that cells over here, these cells, okay, these cells are CD3 negative, CD8 negative. These cells are CD3 positive, but they are CD8 negative. These cells, they are CD3 positive and CD8 positive. And these cells are CD8 positive, but CD3 negative. So these type of segments, each and every type of segments will allow you to identify which type of cells you're dealing with. If you have a high intensity over here, if you have a very high intensity over here, then maximum types of cells in this patient sample are CD3 and CD8. Okay, if you have a very high, for example, once again, if you have a very high intensity over here instead of this one, then these patients, they have CD3 negative and CD8 negative. And this is the method by how you use flow cytometry to identify the different types of CD cells. So for example, over here, cells in the left lower quadrant are both negative for CD3 and CD8. Cells in the right lower quadrant are for CD8 positive and CD3 negative. And then vice versa. Once again, over here, the cells are CD3 positive, CD8 negative. Over here, they're both positive for CD8 and CD3 cells. That's exactly what's written over here. So by using this type of histograms, you can identify which type of antigens are present in which type of cells. So this is used in workup of hematologic abnormalities, for example, leukemia, paroxysmal nocturnal hemoglobinuria, fetal RBCs, and immunodeficiency. So what you will do is write down over here, flow cytometry, and function is detection of hematological abnormalities or malignancies. Okay, this is the way how you can identify which cells are CD3 cells, which cells are CD8 cells, which cells are CD4 cells, and this is how you use flow cytometry in a clinical setting. Are we clear about this, yes or no? Yes or no? Are we clear about this? Are we clear about flow cytometry? Can you guys hear my voice? Are we clear about flow cytometry? Malignancies, hematological abnormalities or hematological malignancies. Okay. Can you repeat the procedure? Sure. So what happens is over here, what the procedure is actually very simple. This is your sample cell. You do not know whether the sample cell has a specific type of antigen. Okay. So what you do is you add an antibody with labeled of fluorescence. Then you add the cells and the then you add the cells and the antibodies together and you let them bind. If there is any binding, you let them bind. After you let them bind, the cells, they undergo through this flow. And over here, you have a laser, which is used to detect one cell at a time. So if there is any type of binding, then what this will do is this will produce this type of histograms over here. For example, if you want to detect a CD8 antigen, you add a CD8 antibody. So over here, if there's high amount of CD8, that CD8 antibody, if you have a patient sample with high amount of CD8 antigens and you add a CD8 antibody, over here, you will see that there's a high amount of intensity of fluorescent scatter in the histogram. And this will help you identify that this patient has a lot of CD8 cells. If you have a lot of intensity over here, then none of these patients have CD8 or CD3. If you have a lot of intensity in this segment, then there's a high amount of CD3 and no CD8. And over here is high CD8 and no CD3. So basically, this is how you identify different types of cellular, for example, hematological antigens by the help of flow cytometry. You add a fluorescent labeled antibody, then you undergo then you let the antibody antigen go through the procedure of flow cytometry. You use the laser to detect the amount of binding, and then you plot a histogram. As a matter of fact, the histograms are not plotted by you, they are plotted by the detector itself. The detector will plot the histogram, and this process is known as flow cytometry. Are we clear about this? Yes or no? Okay. So in order for you to detect any hematological abnormalities or malignancies, you use flow cytometry to detect the problems. That's that. Okay. Next one. Next one is microarrays. Microarrays. What is microarrays? Thousands of nucleic acid sequences are arranged in grids on glass. So you have a lot of nucleic acids which are arranged on grids. DNA or RNA probes are hybridized to the chip, and a scanner will detect the relative amount of complementary binding. This is used to profile gene expression levels of thousands of genes simultaneously to study certain diseases and treatments. They're able to detect single nucleotide polymorphisms and copy number variations for a variety of applications, including genotype, clinical gene testing, forensic analysis, cancer mutations, and genetic linkage analysis. Basically, what microarray is one of the most simplest procedures out of every one of these ones. You have a sample of thousands of nucleic acids. Okay, you have a sample of thousands of nucleic acids. So over here right now, you do not know whether this patient has a certain disease or not, right? So what you do is you add DNA or RNA, which is complementary. For example, let's say you add a DNA which is complementary to a certain cancer. Okay, so for example, you add a complementary DNA. For example, you add GAC. GAC is a complementary DNA for, let's say, liver cancer. For example, for liver cancer. Then you add this DNA, cDNA over here, right? And you wait for this cDNA, this complementary strand, to bind. If there's any nucleotide over here, which is, for example, CTG, right? Meaning ATCG, right? So G will bind to C, T will bind to A, and G will bind to C. If you have any nucleotide sequence, for example, CTG over here, this one will go and bind to this one. And depending on how many of the complementary DNA will bind to CTG will help you identify that whether this patient has a likelihood of developing liver cancer or not. Okay, so this substrate is the complementary DNA or RNA. Okay, so what happens is over here, this is used to see how much of the genetic expression of CTG is present. For example, if there's a high amount of binding or a lot of GAC binding to CTGs because there are lots of CTGs over here, then you can identify with the help of microarrays that there's increased gene expression and the high possibility that this patient will have liver cancer in the future. So over here, let me make another table. Let me make another table. Okay, so once again, name, function. Name is microarray, microarrays. Function is detection of gene expression. Detection of gene expression. Okay. Which procedure will you use if you have to identify cellular size and granularity? Cellular size and granularity. Which procedure will you use? Very good. Cellular size and granularity. If you want to identify size and granularity and protein expression, then you will use flow cytometry. Flow cytometry is hematological abnormalities. Along with that, cellular size, cellular granularity, and protein expression. If you want to identify protein expression, you can use flow cytometry. If you want to identify genetic expressions and the possibilities of the proteins forming in the future, but they have not formed yet, then you can use microarrays. Are we clear about this? Yes or no? Yes or no? Repeat. Okay. Once again, what is microarray? Microarray is basically you have a sample of thousands of nucleotides. So you want to detect the amount of, for example, CTGs in the nucleic acids. So what you do is if you want to detect the amount of CTG, you add a complementary DNA or RNA with the nucleotide sequence of GAC. Okay, for example, you add GAC. And then after there's binding, if there is a lot of binding and then there's a lot of GAC and CTG binding, this will help you to identify that this patient sample have a lot of CTG nucleotides, or the CTG nucleotides are binding really quickly, meaning that they're very potent. Okay, so what happens is if there is a lot of binding, with the complementary DNA to the nucleic acids, then this will help you identify that this patient has increased genetic expression of CTG. And we all know that CTG is our example for liver cancer, for example. So we can conclude that in the future, there's a possibility that this patient has a high likelihood of developing liver cancer. And the procedure which you used is microarray. That's that. Are we clear about this? Yes or no? Okay. Last function of flow cytometry is to assess the cell size, granularity, and protein expressions of protein. Okay. Okay. Next one is ELISA. ELISA is very easy to understand. Enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay, ELISA, is used to detect either an antigen or an antibody. This is an immunological test which is used to detect the presence of either a specific antigen or an antibody. The direct question which you will receive in UWorld is, which procedure will you use to detect antigen and antibody in a clinical setting? For example, you have a patient with a viral infection, for example, hepatitis. You want to detect the patients if they have antigens which are positive for hepatitis or antibodies which are positive for hepatitis. If you want to detect IgG or IgMs, right? And if, for example, we have a patient with hepatitis A, IgM antibodies, then is that an acute infection or is that a chronic infection? Fast answers, please. IgM. If there's a patient who is positive for hepatitis IgM, is it an acute or is it a chronic infection? It's an acute infection, right? And the procedure that you use to identify this IgM is ELISA over here. So the number one function of ELISA is detection of antigen and antibody. The only question you will receive from over here is that the name of the ELISA, and the way that you will be asked the question is, you will have a patient with viral infections, and they want to detect the antibody, whether the patient is positive for the viral infection by detecting the IgM or IgG. If the patient is positive for IgM, then it's an acute infection. If the patient is positive for IgG, then there was a past infection or vaccination. So, and the lab procedure which you use to detect these type of antigens or antibodies is ELISA. Now, ELISA is an immunological test which is used to detect the antigen or antibody in a patient's blood sample. The detection involves the use of an antibody linked to an enzyme. The substrate will react with the enzyme, and this will produce a detectable signal. So, for example, you have hepatitis A antigen, you have hepatitis A antigen, and you add an antibody. For example, this patient is positive for hepatitis, but we don't know that, right? So you want to detect hepatitis. So what you do is you add a hepatitis A antibody. You add a hepatitis A antibody. If the patient is positive for hepatitis A, they will have hepatitis A antigen, and the antibody will go and bind to the antigen. After the antigen and antibody bind, for example, let's say that this is the antigen and this is the antibody. Okay. After they go and bind, on top of the antibody, you have a substrate. Okay, you have a substrate. This substrate has a piece over here which will bind to the enzyme. If there is an enzyme, they will bind. The enzyme that will be added for this procedure will go bind over here after there's binding of antigen antibody, and this enzyme that is added will go and bind over here. Okay. If the enzyme goes and bind over here, then this results in breakdown of the substrate, and this will produce a fluorescence. This will produce a fluorescence. So this will result in this antigen and antibody glow up. So this glowing up of antigen antibody will help the researcher identify that this patient has antigen that is positive for hepatitis A. And this is the procedure of enzyme-linked immunosorbent assay. This is the way you can detect IgG, IgM, and that's exactly how it works. They have a high sensitivity and specificity, but it is less specific than Western blot. For example, if you want to do a Western blot, which can directly detect the amount, the protein, right? So it is less specific than Western blot, but it is very widely used. It's very widely used because it's a very fast procedure. Once again, if the patient has hepatitis A antigen, the antibody will go and bind to the antigen. Once there is binding, the enzyme will go and bind to the substrate, and breaking down of the substrate will result in fluorescence of this antigen-antibody complex. And this fluorescence of the antigen-antibody complex will result in the detection of the antigen or the antibody. Are we clear about this? This is Eliza. The only questions you will receive from Eliza is which procedure do we use in a clinical setting in order to detect the antigen and antibody? The procedure is Eliza. That is this question over here, detection of antigen and antibody. Are we clear about this, yes or no? Okay. Everyone? Okay. Next one is karyotyping. Karyotyping is very easy to understand. So basically karyotyping is, it's a procedure where you can identify the chromosomes, right? So what happens is you add colchicine, which is added to the cultured cells to halt the chromosomes in metaphase. Because we all know that in metaphase, you have the formations of the spindle fibers, and then you have cytokinesis and all those procedures. So colchicine will block the microtubular movements. So you can block the chromosomal, you can block the procedure of division in the phase of metaphase by the help of the drug colchicine. After you stop the procedure in metaphase, the chromosomes are stained, ordered, and numbered according to the morphology, size, and banding pattern. Now, none of them are high yield, except colchicine is added. And at which phase is colchicine added? The colchicine is added at metaphase. This is a question in UWorld. Okay, next one. This can be performed on a sample of blood, bone marrow, amniotic fluid, or placental tissue. So you can perform basically these things where there's high amount of DNA, right? So in the blood, bone marrow, amniotic fluid, or placental tissue, these type of procedures will go very fine. After you do the procedure, you have this type of a blotting, right? And then you can use this type of the blottings to diagnose chromosomal imbalances, autosomal trisomies, sex chromosomal disorders. For example, over here in this patient, you have a certain type of problem. Can anyone identify this? Okay, so they already have the arrows in A, for example. They point to extensive abnormalities in a cancer cells. They have extensive chromosomes over here, okay? Then, for example, let's say that this is 16, 17, 18, 19, 20, 21. Over here, instead of 2 chromosomes, if you had another chromosome over here, then this would be a picture for Down syndrome. If you had another one over here, okay, if you had, for example, this one, instead of the size being like this, let's say the size was now like this, then this would be a picture for Huntington's. Okay, then what else? Then over here, if you had any other problem, for example, you had one of the chromosomes which are deleted, then you can have some other type of problem. So basically, this is the type of procedure by you can identify chromosomal abnormalities and imbalances. So karyotyping is used to diagnose chromosomal imbalances. That is autosomal trisomy, sex chromosomal disorders, and etc. Okay, and the questions are, what type of procedure is used to diagnose chromosomal imbalances? And the answer is karyotyping. Another one is, at which phase is colchicine added? Colchicine is added at metaphase. That is, these are the 2 questions. Okay, is everyone clear about this? Yes or no? Is everyone clear about this? Okay, good. Next one. Next one is fish. Fish is fluorescence in situ hybridization. A fluorescent DNA or RNA probe will bind to specific gene site of interest on chromosomes. Okay. For example, the arrow in A, this arrow, point to abnormalities in a cancer cell whose karyotype is seen above. Okay. For example, over here, that you have the karyotype for the cancer cell, and they're talking about a carcinoma at chromosome number 5. Do you guys have any idea or any diagnosis for a carcinoma at chromosome number 5? We talked about this before. What carcinoma can arise from a chromosome number 5 abnormality? Does anyone remember? Anyone? Any carcinoma from chromosome number 5? Fast answers, please. Yes or no? What is the carcinoma? Very good. Familial adenomatous polyposis, APC at chromosome number 5. Familial adenomatous polyposis. So over here, you have Lynch syndrome is not the one. Lynch is not the one. It's FAP, familial adenomatous polyposis. The arrow in A will point to an abnormality in a cancer cell whose karyotype is seen above. Each fluorescent color represents a chromosome-specific probe. Chromosome-specific probe. It's used for specific localization of genes and direct visualization of chromosomal anomalies. So basically, fluorescent in situ hybridization is the same thing as karyotyping, except the fact that this fluorescent in situ hybridization will help you identify the locations of the genes and visualization of chromosomal anomalies. So basically, you can have microdeletion, translocation, and duplication at 3 levels. You can identify the chromosomal anomalies. And microdeletion, if you have microdeletion, then there will be no fluorescence on a chromosome compared to fluorescence at the same locus on the second copy of that chromosome. So if you have microdeletion, for example, have you guys heard of Cre-du-Chat syndrome? Cre-du-Chat? You have microdeletions in Cre-du-Chat. Has anyone heard of Cre-du-Chat over here? Right. So we will talk about Cre-du-Chat syndrome in a while, but in a Cre-du-Chat, you have micro deletion of a chromosome, meaning that one of the chromosomes are not working or it's not there. So if it's not there, will there be fluorescence? Yes or no? Will there be fluorescence? No. And another one is translocation. Translocation is the fluorescent signal that corresponds to one chromosome is found in a different chromosomes. So for example, 2 white arrows that A shows of chromosome number 17, for example, over here in chromosome number 17, okay, they have translocated to chromosome number 19. Do you realize how chromosome number 17, they have pink, they have a very pink color, right? Chromosome number 19 should not have had any pink color, but this translocation of chromosome from 17 to 19 is showing with the help of fluorescent in situ hybridization. But if you did a karyotyping, only karyotyping, then you could not see the translocations, can you? Yes or no? Could you have seen the translocations in only karyotyping if there was no fluorescence? No. But with the help of fluorescence in situ hybridization, you can identify the translocation. So can we say that if you want to identify the BCR-ABL translocation of the famous leukemia of which is CML, which between chromosome number 9 and 22, can we use fluorescence in situ hybridization to identify the leukemia? Yes or no? Can we do karyotyping to identify the leukemia? No. Okay, good. So that's that. Thank you for understanding that. Next one is duplication. Duplication is a second copy of a chromosome resulting in a trisomy or a tetrasomy. Over here, the 2 blue arrows show duplicated chromosome 8. Over here in chromosome number 8, you see how you have 2 blue arrows. But if there was another color for this 2 chromosomes, for example, if these 2 were blue, then would that be a duplication or would that be a translocation? Fast answers, please. Would that be a duplication or would that be a translocation? It would have been a translocation. And the only way to identify this is fluorescent in situ hybridization. So once again, over here, fluorescent in situ hybridization or FISH. What is the function of FISH? FISH is used to identify specific location of genes and direct visualization of chromosomes. Specific locations of genes and direct visualizations of chromosomes. Are we clear about this, yes or no? Okay. Okay, next one. Once again, fluorescent in situ hybridization is basically karyotyping, but over here you use a fluorescent DNA probe to illuminate the chromosomes. This will help you identify the colors of different types of chromosomes. So in order, for example, karyotyping will allow you to see if there is any change if there is any abnormality, for example, presence or absence of chromosomes, or if there's any type of extra chromosome that is there or a chromosome which is not there, for example, a quantitative way. Okay, so this will help you identify with the help of karyotyping. If you do a fluorescence, you can see if there's translocation, duplication, or absence, or microdeletions. So that's that. Basically, fluorescence is the illuminated form of karyotyping. Okay. Okay. Next one. Can you read again the function you wrote? Okay. The function is specific detection of specific localization of genes and direct visualization of chromosomes. Direct visualization of chromosomes. Okay. Next one. Next one is molecular cloning. Next one is molecular cloning. Molecular cloning is, this is a production of a recombinant DNA molecule in a bacterial host. Okay, this is not very high yield, but molecular cloning is basically, you want to produce a DNA in a bacterial host. So if you want to produce a DNA from a bacterial host, the steps are basically you will isolate an mRNA. My question is, if you want to detect the amount of mRNA over here, which type of procedure will you use? Fast answers, please. If you want to detect the amount of mRNA over here, which type of procedure will you use? Fast answer. Very good. Whenever you hear mRNA detection, the thing I want you to think about is RT-PCR. Okay, step. Let's go back to our text. The first step is isolation of eukaryotic mRNA of interest. So you isolate the mRNA, then you add reverse transcriptase, which is an RNA-dependent DNA polymerase to produce complementary DNA. So you produce DNA from the mRNA. Then you insert the DNA fragments into a bacterial plasmid, and for example, the plasmids will contain the antibiotic resistant genes, and then you transform the recombinant plasmid into a bacteria. After that, the bacteria on the antibiotic medium will produce the cDNAs. Step by step, once again, what's happening over here? You have mRNA. From the mRNA, you have DNA, right? You have the DNA. You have the DNA with the help of reverse transcriptase. Then you add the DNA to a bacterial plasmid, and the bacterial plasmid will now multiply, okay? And now the bacterial plasmids will now multiply with the bacteria, producing extreme amount of the DNA you wanted. This is molecular cloning, step by step, very easy to understand. Okay, this is not very high yield, but the steps are there for your understanding, okay, for your future research purposes. So that's that. This is basically molecular cloning. Are we clear about this, yes or no? Molecular cloning? No. Okay, once again, you have an mRNA, right? From the mRNA, you produce a DNA with the help of reverse transcriptase and enzyme. Then you add the DNA to a bacteria, which has the possibility of multiplication, right? That you insert the DNA fragments into the bacterial plasmids, which will contain the antibiotic resistance genes, because this bacteria, in order for them to multiply, they cannot be killed off by the antibiotic, right? So the plasmids, they have to contain antibiotic resistant genes. So when they multiply, you have the multiplication of the DNA which you wanted. So this is the procedure of molecular cloning. Are we clear now? Yes or no? Okay. Very easy to understand and not very high yield. Do not worry about this. Okay. Okay. Now we will talk about gene expressions and modifications. Gene expressions and modifications. Okay. This part of the lecture is not very high yield at all, But there are certain things which you will be tested on, and I will tell you exactly what you need and exactly what you don't need. So you guys have nothing to worry about. So gene expressions and modifications are transgenic strategies in mice. They involve random insertion of a gene or targeted insertion or deletion of a gene through homologous recombination with the mouse gene. So either you can add a gene or either you can remove a gene. The terms are knock-in. Knock-in is insertion and knock-out is removal. Where did we study knock-in and knock-out? Where did we study knock-in and knock-out? Very good, Crispr Cas9. In Crispr Cas9, we studied knock-in and knock-out. Okay, if it's a random insertion, it's called a constitutive expression. If it's a targeted insertion, it's called a conditional expression, not high yield. Don't worry about this. But if you have to understand, for example, if you want the DNA to be randomly, if you want the gene to be randomly inserted into the DNA, which you're not sure about, the term is constitutive. If you want to target the local location of addition of the nucleotide, then the term is conditional expression. That's that. Next one is Krillok system. Krillok system is, it can inducibly manipulate genes at specific developmental points. Example, to study a gene whose deletion causes embryonic death. So you want to study, you want to use a system which can inducibly manipulate genes at specific developmental points. So what this will do is this will help you identify a gene whose deletion can cause embryonic death. So this is basically a system where you can manipulate. For example, you can use this system. This system is a very complicated system, and it's beyond the scope of step 1. But by the use of Krillok system, you can manipulate the insertion of a gene at any specific location you want. Okay, that's exactly what you need to know. So if you want to insert a gene at chromosome number, for example, 10 at the P sequence, and it's very specific, and that's the exact point where you want to insert the gene, then the system which you use is Krillok's system, Okay, that's that. Once again, not very high yield. Next one is RNA interference, process whereby small non-coding RNA molecules will target mRNAs to inhibit gene expression. So what you do is now you can use Krillok's system to insert a gene at a specific location, or you can use RNA interference by inhibiting genetic expression. So these are the two ways, vice versa, which you can use. One will insert, another one will stop genetic expression. RNA interference, it consists of microRNA and siRNA. Do you guys remember when we talked about microRNAs, we talked about the fact how microRNAs will prevent mRNA function? Yes or no? When we talked about microRNA, right? So microRNAs, they are naturally produced by the cell as hairpin structures. This is high yield. How can you detect microRNA? You can detect microRNA as hairpin structures. This is one question you will receive from this whole thing. Loose nucleotide pairing allows broader targeting of related mRNAs, blocking translation and accelerating mRNA degradation. So basically microRNA, they are used for mRNA degradation. This is another high yield question you will receive. Which structure is responsible for degradation of mRNA? The answer is microRNA. Which structure has a hairpin-like appearance? The answer is microRNA. That's that. Abnormal expression of microRNAs can contribute to certain malignancies, for example, because if you have a lot of microRNAs, then that can prevent the mRNAs, which is responsible for production of proteins that can prevent cancers. So if you do not have those mRNAs, because you have a lot of those microRNAs which are destroying the mRNAs, then you can have certain malignancies. Okay, so that's that. Next one is siRNA. Small interfering RNAs, they are usually derived from exogenous double-stranded source. Once inside a cell, the siRNA requires complete nucleotide pairing, leading to a highly specific mRNA targeting. This results in mRNA cleavage prior to translocation. So siRNA will break down the mRNA before the mRNA could translate. So once again, siRNA is also responsible for breaking down mRNAs. So that's that. But
siRNAs, they can be produced in vitro transcription for knockdown experiments. For example, if you want to prevent a protein to be formed from an mRNA, you cannot create your own microRNA. Okay, they are naturally produced, but you can come up with your own siRNA, which will interfere with the mRNA to form the protein by breaking down the mRNA before it can translate into a protein structure. So that's that. The only two questions you will receive from over here is hairpin structure, which is microRNA, and which structure is naturally produced to break down mRNA, that is microRNA, and which structure is produced in vitro to break down the mRNA, that is siRNA. One is naturally produced, another one is produced in vitro. That's that. Are we clear about this? Yes or no? Okay, once again, you have two structures for breaking down of mRNA. One is microRNA, another one is siRNA. MicroRNA is produced naturally. siRNA is produced in vitro. Okay? Yes. So we prevent protein from producing by breaking down mRNA. Yes. If it's by miRNA, then it's a natural production. There's nothing we can do. But if you want to perform an experiment, then you can come up with siRNA, which will prevent mRNA transcription. Are we clear about this? Yes or no? And with that being said, you are done with the laboratory procedures. Okay, okay, so right now, if I ask you, has this lecture helped you, has this lecture simplified the whole thing, the whole lab procedures, yes or no? Have you guys understood the lab procedures in a very simplified form? Number one, if the question is yes, then thank you. My next question is, have you guys understood exactly the types of question you will receive from the lab procedures? Basically the names and the functions, okay? Are we clear about this? Yes or no? Molecular cloning is not high yield, that's why I didn't add it. Okay, molecular cloning is not very high yield, so that's why I did not add molecular cloning, okay? So if you want to do a quick revision of lab procedures, you can just go through the notes, this notes over here, and this will help you identify all the high yield questions, okay? Are we clear about this? Okay. So with that being said, can we take a small break before we jump into the genetic terms? Yes? Okay. All right, so let's take our break. Let's come back in 10 to 15 minutes. Okay. Okay, is everyone back from their break? Can you guys hear my voice? Okay. Do we understand the lab procedures perfectly? And if we receive questions from them, can we do them with absolute perfection? Okay. Let me do a small revision to see if you guys have understood, right? Can we do the revision for like 2 minutes to see if we have understood the questions which we will get from the lab procedures and techniques? Okay. Which type of procedure will you use for amplification of a piece of a DNA so that you can identify if that piece of DNA is for a certain individual? Okay, which procedure will you perform to detect mRNA? Good. Which procedure will you perform in order to remove a part of DNA and add another healthy DNA? Okay. Okay, which procedure will you use to identify C-Jun and C-Phos? C-Jun and C-Phos. Crispy. Okay. It's CRISPR. when you say crispy, it sounds like, okay. Crispy is for fried chicken. Okay. CRISPR is your genome editing tool. Autocorrect, no problem. I was just kidding. I was joking. No problem. I understand exactly that you wanted to mean CRISPR. No problem at all. What blotting procedure will you use to identify RNA? Proteins? Which procedure will you use to identify cellular size and granularity? Cellular size and granularity. Which procedure will you use to identify gene expression, the amount of gene expression? Gene expression. Microarray, very good. Microarray. Which procedure will you use to identify antigens and antibody of a disease? Eliza. Which procedure will you use to identify genetic translocation? Karyotyping is not the correct answer. FISH is the correct answer. Which procedure will you use to identify chromosomal disorders? Okay. Which structure is produced naturally to prevent mRNA transcription? Which structure is produced in vitro to prevent mRNA transcription? Okay. Are we clear about this? The lab procedures, the lab techniques? Okay, I can assure you that if you can remember these informations, because you have to understand that when you give your USMLE step 1, you will have to carry a ton of information to your exam, right? So from these lab procedures and techniques, these are the only piece of information that is extremely high yield, so that you can remember these informations exactly to do well in the actual exam. Okay, so if you want to concise all the informations, then the questions which I just asked you right now is exactly what you need. Of course, there will be some questions which will be a bit in depth. And for those questions, we had that big discussion for at least an hour or so. Okay, if you guys receive questions from lab techniques which are more complicated, then please let me know. I'll try to break it down for you. But I highly doubt you will receive any more question, which is more than what you just studied right now. Okay, so fish is more precise than karyotype. Yes, fish is more precise. Okay, fish is more precise. Okay. So with that being said, can we jump into the genetic terms? Okay. How important are the genetic terms? Okay, the genetic terms are very, very important. The genetic terms are very, very important. Okay, So the genetic terms, okay, Dr. Mohammed, you have nothing to be worried about. We will send you the lecture. You can watch the lecture and let me know if you need help with anything tomorrow, okay? Okay, so let's jump into the genetic terms. First of all, we have codominance. Codominance basically is, okay, so before we talk about the lecture, let me tell you exactly how you will receive your questions. Your questions in USMLE or UWorld will come from the examples, and the answers are there to see whether you have the knowledge to differentiate each and every term from one another, okay? So with that being said, just as how we made a table for lab procedures, we will make another table for the genetic terms to help us identify exactly what we need to review for a fast revision of the first aid. Okay, so with that being said, are we ready with our notebook? Are we ready with our notebook? Okay. Over here, you will write diseases and pathology and example. And over here, you will write terms. Okay. Number one. Number one is codominance. What is codominance? Codominance is the term which is used when both alleles will contribute to the phenotype of a heterozygote. We know that the phenotype is the physical expression of the genes, and genotype is basically the genetic sequence in the DNA, right? So if both alleles are expressed, then this is known as codominance. The example for this one is blood groups A, B, AB, alpha-1 antitrypsin, and HLA groups. People with these type of DNAs, which is almost every one of us except alpha-1 antitrypsin, right? The physiological blood groups are there in everyone. And each and every allele of the blood groups are expressed in humans. So codominance is basically if you have two alleles, right? If you have two alleles, let's say these are two chromosomes, right? You have one allele over here, you have another allele over here. Both of the alleles are now being expressed. Not a single one. If a single one was expressed, then this would have known as a singular genotypical phenotype. Since both of them are expressed over here, this is known as codominance. Codominance, okay? So both alleles will contribute to the phenotype of the heterozygote. This is known as codominance. Once again, over here, the number one example is HLA group. And blood groups A, basically, one second, blood groups. Just write blood groups. And the term is codominance. Okay, the term is codominance. The question you will receive is you will receive some sort of a question in NBME or UWorld where they will talk about the patient's blood group and this and that, and they will try to make the question sound as complicated as possible, and then they will ask you, what is the genetic term used to identify the blood group of the patient? And the term is codominance. Are we clear about this, yes or no? Yes, okay, next one. Next one is variable expressivity. Variable expressivity. Alpha-1 antitrypsin is not high yield. That's why I did not mention. HLA and blood groups are more high yield. Next one is variable expressivity. That is, patients with the same genotype have varying phenotypes, meaning that if there was one type of genotype which was responsible for a certain phenotype, then that would not have been variable expressivity. But patients with same type of genes which have different types of physical appearance is known as variable expressivity. The number one disease where variable expressivity is used is neurofibromatosis type 1, NF1. NF1 is used for variable expressivity. Over here in NF1, the genes, there are different types of genes over here which will give rise to the neurofibromas. But for example, if in a normal patient, in a patient who does not have neurofibromatosis, the genes are there but the phenotype is not expressed. But in a patient of neurofibromatosis, the genes are there and the genes are expressed in the physical appearance of the neurofibromas. So number 2 is neurofibromatosis, number 1 type of example, and the term is variable expressivity. The question once again might be about neurofibromatosis. Exactly, since USMLE has a tendency of making questions exactly from the first aid. So they will talk about neurofibromatosis and they will talk about what is the genetic term used to identify neurofibromatosis, and the genetic term is variable expressivity. That's that. Okay, next one. Are we clear about variable expressivity? Next one. Next one is incomplete penetrance. Incomplete penetrance meaning not all individuals with a mutant genotype show the mutant phenotype, meaning that once again, you have some sort of a genetic mutation. Let's say in John and Jane, okay? John has 2 mutant genes. Jane also has 2 mutant genes, okay? But in John, the mutant genes are not expressed. In Jane, the mutant genes are expressed. As a result, John does not have the phenotype. Jane has the phenotype, as simple as that. The fact that the fact that not all individuals with a mutant genotype will show the mutant phenotype, this is known as incomplete penetrance. My apologies, I'm sorry. For example, BRCA1 mutations do not always result in breast or ovarian carcinomas over here, right? So for example, if John does not have the expression of the breast or ovarian carcinoma, right? Then John has incomplete penetrance. If Jane has the mutant genes, but Jane has if Jane has the mutant genes and Jane also has the carcinoma, then Jane has complete penetrance. Since John has the mutant genes, but John does not have the phenotype, John is an example for incomplete penetrance. Over here, the example for incomplete penetrance is BRCA1 and ovarian carcinoma. The term is incomplete penetrance. Are we clear about this? Yes or no? Are we clear about this? Incomplete penetrance? Okay. So we will read 5 at a time. We will read 5 at a time. Okay. Next one. Next one is pleiotropy. What is pleiotropy? Pleiotropy is one gene will contribute to multiple phenotypes. For example, if you had one gene which can give rise to a lot of different types of physical expressions, this is known as pleiotropy. Pleiotropy means plural genes. Plural genes. Plural physical appearance for one gene. Plural genes is not the right thing to say. Plural phenotypes for one genotype. So plural phenotype for one genotype is pleiotropy. Pleiotropy, plural phenotype for one type of gene. So one gene can contribute to multiple phenotype. Example is phenylketonuria, will manifest with light skin, intellectual disability, and musty body odor. For example, the single gene which can give rise to phenylketonuria is also responsible for the development of the light skin, as we see in phenylketonuria, intellectual disability, and musty body odor. So over here, you will write phenylketonuria, and this is pleiotropy. Pleiotropy. Okay. Next one. Next one is anticipation. Anticipation is extremely easy to understand. Anticipation is basically with increasing severity or earlier onset of disease in succeeding generations, meaning that if you have a patient who developed Huntington's disease at 50 years of age, the patient's son can develop the disease at 30 years of age, and his son can develop the disease at 12 years of age. Until and unless an entire generation is wiped out by Huntington's disease, this process is known as anticipation. Because as you know, if you have a young patient at 12, for example, they develop Huntington's disease, which is extremely rare, then this patient has a high rate of mortality and morbidity, and they will pass away without producing another generation. So this generation is wiped out. So if once again, if a patient has Huntington's at 50, his son can have the Huntington's at 30, and his son can have Huntington's at 12, so this and that. So the severity of the trinucleotide repeats or the severity of the disease will increase in succeeding generation. And in every trinucleotide repeat disorder, anticipation is there. So for this one, we will write trinucleotide repeat disorders. And the genetic term is anticipation. Okay, anticipation. I'll give you guys exactly 2 minutes. And after 2 minutes, I will ask you the terms and which disease is associated with which term. So this is the part of the table which you have to recap, memorize, and keep in mind. And this is the question. This is the part of the table where I will ask you the questions from. Okay. This is the type of practice I want you guys to have when you do the revision and recapitulation, because this is exactly the same way you will receive your questions in your actual exam. And believe me or not, these are the same exact examples which you will get for the UWorld, because they will try to make the questions easy by mentioning exactly the diseases from first aid. So this will go hand in hand. You have 2 minutes in your hand. It's 11:22. I'll ask you the questions at exactly at 11:22. I'm sorry, at 11:23, and see whether you guys have understood or not. Okay, so 2 minutes on the clock. Let's do this. Okay, is everyone ready? Is everyone ready? Okay. So what is the term that is used for HLA and blood groups? HLA and blood groups. Okay. What is the term used for phenylketonuria? Phenylketonuria. What is the blood group used for trinucleotide repeat disorders? Okay. What is the blood group used for, what is the term used for neurofibromatosis? What is the term used for BRCA1 and ovarian carcinoma? Okay. Incomplete penetrance. Okay. Since this is something which would require you to look over time and time again, because there's a possibility you will forget this after the lecture is over, I need you guys to take a picture of this either in your phone or your laptop, or print it out and post it on your wall so that every day you can go through them. And this will help you remember all the terms with absolute perfection. Okay, next one. Next one is the loss of heterozygosity. Loss of heterozygosity. Loss of heterozygosity is if a patient inherits or develops a mutation in a tumor suppressor gene. Okay, if a patient inherits or develops a mutation in a tumor suppressor gene, the complementary allele must be deleted or mutated before the cancer develops. Once again, if a patient inherits or develops a mutation in a tumor suppressor gene, you have tumor suppressor genes um, which are responsible for suppressing the excessive growth of genes, right? And if you have a mutation in these type of genes, the complementary allele must be deleted or mutated before the cancer can develop. So in order for the cancer to develop, the tumor suppressor gene have to be knocked out. And the process of knocking out of tumor suppressor genes, is by either mutating a part of the tumor suppressor gene or deleting a part of the tumor suppressor gene. That is that. The famous examples for loss of heterozygosity is retinoblastoma, or the two-hit hypothesis, Lynch syndrome over here. So over here, we have retinoblastoma and Lynch syndrome. And in both of these cases, the genetic term is loss of heterozygosity. Once again, heterozygosity means that there could be two genes, right? And if you have a mutation in one gene, then this could result in no changes in the tumor suppressor gene, right? But if there's a mutation in both of these genes, then this gene will get knocked out. If this gene gets knocked out, then the tumor suppressor genes will not be working. If they do not work, then cancers such as retinoblastoma and Lynch syndrome could develop. And this is known as loss of heterozygosity. Okay, that's that. Next one. Has everyone understood loss of heterozygosity? Yes. Two-hit hypothesis, meaning both of the genes are knocked out in Lynch syndrome. Both genes are knocked out in Lynch syndrome. Okay. Next one. Next one is dominant negative mutation. Dominant negative mutations. Students with 2019 first aid, do you guys have dominant negative mutations? Yes? Okay, good. Dominant negative mutation. Basically, this is a mutation which will exert a dominant effect. A heterozygote will produce a non-functional protein that also prevents the normal gene product from functioning, okay? So what happens is, over here, you can have two heterozygote genes. These two heterozygote genes, right? And these two genes in a chromosome, they can result in the production of a non-functional protein. The protein will not function, and this protein will prevent the normal functioning of the gene. So the protein will prevent the gene from functioning. Over here, the best example is P53 tumor suppressor genes results in a protein that is able to bind DNA and block the P53 from binding to the promoter. So over here, if you have a P53 tumor suppressor gene, which is mutated, this can result in the formation of a protein which will prevent the further action of the P53. So if there is any gene which can result in the formation of a protein which will prevent its own function, this term is known as dominant negative mutation. This term is known as dominant negative mutation. So once again, our example, disease or pathology, and we have terms. And the example is P53 mutation. P53 mutation is our example for the fact that if there's a P53 mutations, you can have a DNA binding protein which can prevent the P53 from binding in the future and prevent the action of the P53. The term is known as dominant negative mutation. Okay. Once again, if you have any gene which can give rise to a protein that can prevent the action of that gene in the future, this is known as dominant negative mutation. P53 mutation results in the formation of a DNA binding protein which can prevent the action of the P53 in the future. And this is the term that is best used for the example of P53 mutation, that is dominant negative mutation. Are we clear about this? Yes or no? Any mutation resulting in the formation of a protein which prevents the future action of the gene is known as dominant negative. Dominant negative. Dominant positive, meaning that the gene will produce a certain phenotype that will work for the gene, and the gene can keep on producing the type of proteins they are responsible for, for which they are responsible. But if the gene results in the formation of a single type of protein, which prevents further action of the gene, this is known as dominant negative. Think of this as like a negative feedback to gene function. Okay, think of this as a negative feedback to gene function. Okay. No, normally, do we have negative feedback in genetic function? Is that normal or abnormal? Do we have any negative feedback in genetic function? Can excessive production of a protein prevent gene expression? Yes or no? Can excessive production of a protein prevent gene expression? No. No, that is abnormal. If excessive production of a protein prevents gene expression or functioning of the gene, this is known as dominant negative mutation. Okay. Are we clear about this? Silencer is not the one. Okay, once again, you have a chromosome. Over here, you have genes. They are forming the proteins. Okay, proteins are happy, genes are happy. Now you have a chromosome with a gene that is forming a protein that is angry. And this protein prevents the function of this gene. This is known as dominant negative mutation. Are we clear about this? Yes or no? Okay. Okay. Next one. Next one is linkage disequilibrium. What is linkage disequilibrium? Linkage disequilibrium is the tendency for certain alleles at two linked loci to occur together more or less often than expected by chance. So it's a tendency for the alleles at two linked loci to occur together. For example, this is measured in a population, not in a family, and often varies in different populations, meaning that, for example, if you have this chromosome and this chromosome, if there was one gene or one allele over here and this allele over here, okay? For example, right now, if you have this allele, instead of being on this part of this gene, this comes over here throughout the generations, throughout populations. So there is shifting of the alleles. So this is the tendency for the alleles at two linked loci to occur together, more or less expected by chance. So what happens is in the next generation, the alleles will be there in this strand of chromosome instead of being on two different types of chromosomes. So what they do is they break the equilibrium and they form a link. And this tendency of these two genes to stay together at the same loci by chance in a population by breaking the equilibrium is known as linkage disequilibrium. Once again, linkage disequilibrium and dominant negative mutation, they're not very high yield. This is just for your understanding that with subsequent generations, there could be a possibility that the genes could break off from one strand and they could be there in another strand. This is known as linkage disequilibrium. Are we clear about this? Yes or no? Yes or no, linkage disequilibrium? Okay, good. Next one, this is high yield. Mosaicism. Mosaicism is the presence of genetically distinct cell lines in same individual. So there are two types of mosaicism, that is somatic and gonadal. Somatic mosaicism is mutation arises from mitotic errors after fertilization and propagates through multiple tissues or organs. So there can be a mutation which can arise after a mitosis, and this mutation which has happened in the process of mitosis, they can be presented on multiple tissues and or organs. The same thing is if there's a mutation in egg or sperm cells during the process of cellular division, and if that mutation stays in the gonadal cells, if the parents and relatives do not have the disease, then it's known as gonadal mosaicism. Okay. Right now, if I tell you that you have patients of Down syndrome who have babies with Down syndrome, patients of Down syndrome who have babies with Down syndrome, what type of mosaicism is this? Somatic mosaicism or gonadal mosaicism? Fast answers, please. If patients and relatives do not have the disease, if the parents do not have the disease, I'm sorry, not the patients, the parents. If I tell you the parents have Down syndrome and the babies have Down syndrome, this is known as somatic mosaicism because the mutation have arisen and the mutation have stayed in multiple tissues and organs, and this could have given rise to somatic mosaicism. But parents having Down syndrome and babies having Down syndrome is extremely rare. The most common one is gonadal mosaicism. If the parents do not have Down syndrome, but the babies have Down syndrome, then that must mean that the cellular division mutation has occurred only in the sex cells or only in the cells responsible for fertilizations or the gonadal cells. So gonadal mosaicism is the mitotic or the meiotic error or cellular division error, which has happened only in the gonadal cells and which has persisted and given rise to and gave rise to Down syndrome. This is known as gonadal mosaicism. The most common example of mosaicism is McCune-Albright syndrome. This is an entire question by itself. This is very high yield. What is McCune-Albright syndrome? McCune-Albright syndrome is due to GS protein activating mutation. This can present with unilateral café-au-lait spots, polyostotic fibrous dysplasia. We talked about this. The bone is replaced by collagen and fibroblast and at least one endocrinopathy. Did we talk about this once before? Yes or no? I asked you guys about the 3 findings in McCune-Albright syndrome. Do you guys remember? And we had students who mentioned this because they studied first aid before, right? So what is McCune-Albright syndrome? What is McCune-Albright syndrome? McCune-Albright syndrome is basically if you have a triad. There's a triad. The triad is, first of all, you have café-au-lait spots. What is café-au-lait spots? What is café-au-lait spots? Oh, we have a picture. Okay, very good. Picture A. Basically, picture A will show that. Do you guys remember how when you drink coffees or cappuccino, you have there's milk-like spots on top of the coffees? There's milk-like spots on top of the coffees, right? If you have spots like that on your skin, that is known as café-au-lait spots. Café-au-lait spots. If you see patients with café-au-lait spots, can you guys tell me another diagnosis which you can suspect? Fast answers, please. Which you can suspect. Neurofibromatosis. Very good. Neurofibromatosis. So café-au-lait spots. Then you have polyostotic fibrous dysplasia. What is polyostotic fibrous dysplasia? This is basically a very fancy term for the fact that the bones are replaced by collagens. You have collagens which forms bones, but over here you have bones replaced by collagens and fibroblasts. This is known as polyostotic fibrodysplasia. Another one is endocrinopathy, any type of endocrinopathy. This could be Cushing syndrome, Conn syndrome, Addison disease, anything else, anything. This could be absolutely anything. If you have these 3 things in one individual, this is known as McCune-Albright syndrome. This is known as McCune-Albright syndrome. Once again, it's a triad of café-au-lait spot, Polyostotic fibrous dysplasia, meaning bones replaced by collagens, and any type of endocrinopathy. The reason why these three are happening in this individual and forming a syndrome is because there is a mutation in GS protein. GS protein. And this is an example of mosaicism because this mutation occurs in the steps of cellular division. Okay, this mutation occurs in the steps of cellular division. If the mutation occurs in multiple tissues and organs, and what type of mosaicism is this? Somatic mosaicism or gonadal mosaicism? Very good. It is somatic mosaicism. Okay. 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 Next one. Next one is if this type of mutation they occur, if this type of mutation they occur before fertilization, that is before the fertilization has occurred, but this type of mutations are happening, this can be extremely lethal. But if they're happening after the baby is born, then this is this is survivable. The patients can survive. Okay, that's that. Somatic won't affect the baby. High, in most cases, this could or could not affect the baby. That's not the thing which we're focusing on over here. The thing which we're focusing on over here is if the mosaicism happened before fertilization, then it will be lethal. If it happens after fertilization, then it's not lethal, it's survivable. Okay, are we clear about this? Are we clear about this, yes or no? So for our one pager, the diagnosis is oh, right, okay. So over here, for your one pager, this is what you mean. Over here, we have two types of examples. Okay. Over here, you have two types of example. For this one, you have mercury albrite and the mercury albrite, and the term is mosaicism or Down syndrome. Okay, are we clear about this? Yes or no? Okay, next one. Next one is locus heterogeneity or heterogeneity. Locus heterogeneity is mutations at different loci can produce similar phenotype. So before we began our lecture on genetics, we talked about genes, chromosomes, alleles, and locus. Locus are basically the positions of the alleles. If you have mutations at different locations of the alleles, they can still produce a same type of physical expression. This is known as locus heterogeneity. For example, in a patient with albinism, you can have genetic mutations occurring at different types of locations, but at the end of the day, if the mutations are there and at any type of location, the phenotype for albinism will happen regardless. This is known as locus heterogeneity. So over here, for this one, your example is albinism. Your example is albinism. And the term is locus heterogeneity. Okay. Are we clear about this? Yes or no? Okay, next one. Next one is allelic heterogeneity. Different mutations in the same locus can produce the same phenotype. So before we had mutations happening at different locus producing the same type of physical expression. Now we have the mutations at the same locations, which can produce the same type of mutations. Same type of mutation. This is known as beta. This is known as allelic heterogeneity. The example is beta thalassemia. The example is beta thalassemia. Okay, so you have beta thalassemia, and the term is allelic heterogeneity. Once again, the questions you will get from UWorld are directly from the diseases. They will describe these diseases exactly as how they are represented in first aid, and they will ask you which term is responsible for this disease. So if you can remember the examples with the term, then you can answer each and every question from the genetic terms with absolute perfection. Okay, that's that. Allelic heterogeneity. Okay. Can we connect splicing with this regarding to beta thalassemia? That is the pathology. That is not the term. Okay, splicing mutations are the pathology of beta thalassemia. Allelic heterogeneity is the term used to describe beta thalassemia. Okay, terms are not the pathologies. Terms are the genetic terms, are the terms used to describe each and every diseases. Okay, so that's that. Okay, but very good to know that you remember that splicing is responsible for beta thalassemia. So that's that. But over here, allelic heterogeneity is the term. Splicing is the pathology. Okay, so that's that. Are we clear about allelic heterogeneity? Okay. Next one. Next one is heteroplasmy. What is heteroplasmy? Heteroplasmy is the presence of both normal and mutated DNA, which can result in variable expression in mitochondrial inherited disease. So there can be presence of both normal DNA and mutation of mitochondrial DNA. This can result in variable expression in mitochondrial inherited diseases. Basically, that if you have a DNA which has been mutated, especially if you have a DNA in the mitochondria which has been mutated, and this mutated DNA in the mitochondria can get passed on to generations. So this passing on of this passing on of mutated mitochondrial DNA, this term is known as heteroplasmy. So heteroplasmy is basically the mutated mitochondrial DNA passing on from generations to generation, especially from mothers to children, because we know that fathers cannot pass down the mitochondrial DNA. Your mitochondrial DNA comes from your mother. So if there was a mutations in the mother's mitochondrial DNA, that would 100% be passed down to the child if that mutated DNA had heteroplasmy. So that's that. So mitochondrial DNA passed from mother to all children. The examples of this is MELAs. Have you guys heard of MELAs? Yes. Can you guys name another mitochondrial disease? We talked about this in the UWorld notes. Mitochondrial disease, another one. Leber's hereditary optic. Leber's hereditary. Very good. Leber's hereditary optic atrophy. Okay. And the term is heteroplasmy. Okay. Are we clear about this? MELAs. MELAs is myogenic epilepsy-like episodes and stroke-like syndrome. Okay. And MELAs. Okay. Are we clear about this? Okay. Before we jump into uniparental disomy, can I give you guys 5 minutes to read all the terms and then I'll ask you questions? Okay, yes or no? I'll give you guys exactly 5 minutes and then I'll ask you questions from the examples. I'll ask you questions regarding the terms if you guys can remember the terms, and please read the previous ones too, these ones, because it gets very confusing until and unless you have every one of this memorized. Okay, so I'll give you guys 2 minutes. I'm sorry, 5 minutes to go through those terms. After 5 minutes, at exactly 11:55, you wrote labor. Then what's the second thing? Labor's hereditary optic atrophy. Okay. Optic atrophy or optic neuropathy? Anyone? Okay. I'll give you guys exactly 5 minutes. After 5 minutes, I'll ask you guys the terms. Okay, take 5 minutes and learn the terms, please. Okay, is everyone ready? Is everyone ready for the questions? Okay, good. Once again, HLA and blood groups, what is the genetic term? HLA and blood group, what is the genetic term for HLA and blood groups? Very good. What is the genetic term for neurofibromatosis? What is the example for pleiotropy? What is the example for pleiotropy? Very good. What is the example for anticipation? Okay. Retinoblastoma and hereditary non-polyposis coli, or Lee from any syndromes. What is the term? Very good. What is the term for single P53 mutations? P53 mutations, negative, dominant negative effect. Very good. What is the term used for um, Down syndrome? Mosaicism. What is the example? What is the term used for albinism? What is the term used for beta thalassemia? Okay. What is the term used for Meller's and Leber's head hereditary optic atrophy or neuropathy? Very good. What is the diagnosis for a patient with polyostotic fibrous dysplasia, endocrinopathy, and cafe-au-lait spots? What is the mutations in which gene? What is the name of the gene? GS gene. GS gene. Okay. Before we start studying about uniparental disomy and Hardy-Weinberg genetics, can we go through a break of 5 minutes, please? Before we begin the break, have you guys understood all the ways you guys will receive the questions from the genetic terms? Yes or no? Okay, good. Are we confident about the genetic terms? Okay. Okay, so we will come back after 5 minutes and we will talk about Hardy-Weinberg principles and uniparental disomy, and we will also talk about imprinting disorders. Okay, so let's see each other. Let's come back in 5 minutes. Okay, is everyone back from the break? Is everyone back from their break? Okay. Okay. Um, before we talk about uniparental disomy and imprinting, I want to talk about another thing, that is uniparental disomy and imprinting will go hand in hand for two types of diseases, that is Prader-Willi syndrome and Angelman syndrome. Okay, so we will talk about this in 1 second, and we will talk about them together. But before we talk about them, can we talk a little bit about the Hardy-Weinberg population genetics, or the Hardy-Weinberg principles? Can you repeat loss of heterozygosity and linkage disequilibrium? Okay, so loss of heterozygosity is basically if both of the genes are knocked out, and this both knocking out of genes results in the formation of diseases. This is known as loss of heterozygosity. For example, in hereditary non-polyposis coli syndrome, you have two head hypotheses where both of the genes are knocked out. In two both knocked out genes, you have the formation of a disease, which would not have happened if one of the genes was there. So due to loss of both genes of the heterozygote, this loss is known as loss of heterozygosity, and this term is used to describe the disease that is hereditary non-polyposis coli. That's number one. Number two is linkage disequilibrium. Linkage disequilibrium is it's basically the tendency for alleles at two linked loci to come together to come together in subsequent generations, because in the previous generations these two alleles were at different locuses, at different positions, at different chromosomes. Now with subsequent generations, these genes, they have come together at a single linked locus. This is known as linkage disequilibrium. Are we clear about this? Yes or no? Out of linkage disequilibrium and loss of heterozygosity, the most important one is loss of heterozygosity. And the example of loss of heterozygosity is retinoblastoma, hereditary non-polyposis coli, and P53 Li-Fraumeni syndrome. Okay, Li-Fraumeni syndrome. Okay. Are we clear about the types of question? What's an example of linkage disequilibrium? To be honest, the best way you can identify the diseases of linkage disequilibrium is if they have a question where they mention that in previous population the genes were at different locuses. Now in this population the genes are at the linked locus. If they are talking about previously they were at different positions, and now the disease, it could be any rare disease, which will mention that with this disease, you have the genes at two linked locuses. This is known as linkage disequilibrium. So you have to identify the term. You have to identify the term. And then once again, linkage disequilibrium is not that very high yield. But if they talk about any rare disease, once again, if they talk about any rare disease where in the previous population, they were they were at different locations. Now in this population, the disease is occurring because the genes are at linked locuses or at linked loci. Then this is known as linkage disequilibrium. Okay, so that's that. Okay, Hardy-Weinberg principle. Before, okay, Hardy-Weinberg principle. I want to get done with this part because then I can talk about uniparental disomy and imprinting together. Okay, let's talk about Hardy-Weinberg population genetics. This is very, very high yield, and this is a bit complicated, and we will try to incorporate the information with a question, so you have nothing to worry about. Is everyone ready? Can I have your attention? Okay, good. Let's start with the Hardy-Weinberg population genetics. For example, if P and Q represent the frequencies of alleles A and A, that is capital A and small letter a, respectively in a population, then P plus Q equals 1. So basically you have A, right? Over here, the entire population could be said that this is A plus A equals 1. Okay, let me take a blank page. Let me break it down. Okay. What's happening over here is you have A, 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 A for one type of population, right? And then if you draw the entire board, then you have A, then you have A, and this is what it is. If you represent this big letter with P and this small letter with Q, right? Then this entire population, how many types of allele does this population have? Can I say that this entire population has two types of allele, that is A and A? Yes or no? This population has only two alleles. Yes or no? A and A. Okay. So if A is P and if A is Q, then what we can write is the entire population can be said to be 1. So P plus Q equals to 1. This is the derivative of the Hardy-Weinberg equation, Hardy-Weinberg population equation. Okay, P plus Q equals 1. Over here, the P is 4. Okay, the P is for the capital A and Q is for the little a. Okay, right now, if I tell you that, P plus Q equals 1, then we can have another derivative over here, which can also be said that P square plus 2PQ plus Q square equals 1. Okay, so there are two types of Hardy-Weinberg equation, that is P plus Q equals to 1 and P square plus 2PQ plus Q square equals to 1. With this equation, we can say that P square is your frequency of homozygous allele A and Q square is your frequency for the homozygous allele small a. Okay, small a. So over here, basically, you have A, okay, because we know that P square, okay, let me write this again, P square plus 2PQ plus Q square equals to 1. We generally we assume that in a normal patient, the dominant allele will be a healthy allele that is expressed. That is, for example, if you have a normal patient with an allele that is expressed, it's a healthy allele. And with that assumption, we can assume that P square will represent the healthy individuals. Okay, Q square, that's what we assume. Q square is for diseased individual. And 2PQ is our carriers. Okay. Okay. With this, in our knowledge, if I ask you that you have a question where I ask you, for example, there's a couple who wants to know if their child has cystic fibrosis. For example, there's a couple who wants to know if their child has cystic fibrosis, okay? And the disease frequency is in the population. The disease frequency is 1 out of 90,000 people in that population have cystic fibrosis. That is, out of 90,000 people in that population, only one person has cystic fibrosis. So the disease frequency is 1 out of 90,000. The question is, if I tell you that this is the disease frequency, what are the chances of being a carrier. Okay. So if the disease frequency is 1 out of 90,000, can we assume that the healthy frequency is 89,999 out of 90,000? Yes or no? Because we know both of them together will result into 1, right? Okay. So if I tell you that disease frequency is 1 out of 90,000, we know that the diseased allele is Q square. So can we write Q square equals to 1 out of 90,000? This is the disease frequency, because from the previous ones, we wrote that Q square represents the diseased individuals. So can we say Q equals to 1 by 90,000? Yes or no? 1 by 90,000, yes or no? 1 by 90,000 to the square root of 2. And the answer would be 1 by 300. And the answer would be 1 by 300. So Q equals to 1 by 300. If Q equals to 1 by 300, then can we write P equals to 1 minus 1 by 300? Because P plus Q equals to 1, yes or no? Yes. So the answer would be, what is 1 minus 1 by 300? What is 1 minus 1 by 300? Okay. I'm not sure. 299 by 300. Right. So let's 299 by 300. Okay. No, I wanted to know the I know it's 299 by 300. I wanted to know that the decimal. Does anyone have a calculator? I'm sorry, I don't have an access to a calculator. Just write down 299 by 300 and just let me know the value. Let me know the value. 0.99. Okay, good, very good. So 1 minus 1 by 300 is 0.99. Okay. So if P equals to 0.99, then can we assume that our question was, what are the chances of being a carrier? So the carrier frequency is 2PQ. So can we assume that 2 into 0.99 into 1 by 300? And the answer would be, would anyone be kind enough to let me know the answer, please, from the calculator? Anyone? 2.2 into 0.99 into 1 by 300. Should be around close to 1 by 150, 0.006. Very good, thank you. So this is the carrier frequency, and this is your answer. Okay, so if they ask you to identify the carrier frequency, this is how you will do the calculations. Are we clear about this, yes or no? Yes, okay. Have you guys understood the Hardy-Weinberg's equation? The Hardy-Weinberg's equation, yes or no? P squared plus 2PQ plus Q squared. Okay, next one. Next one is cystic fibrosis. What type of a disease is cystic fibrosis? Autosomal recessive. Autosomal recessive means that both of the zygote, both of the genes, they should be for homozygous for the recessive allele, yes or no? Homozygous for the recessive allele, yes? Okay. So, okay. Oops. One second. Okay. So if we want to draw an autosomal recessive one, so let's say that this is A over here once again. Okay. Again, A, and then A, and then A. Okay. Okay. Now, my question is, what is the possibility of over here? If I say 1, 2, 3, 4, which number is the diseased individual? Which number is the diseased individual? 4. So 4 is the diseased. Okay. Okay. Which numbers are not the diseased individuals? 1, 2, and 3, right? 1, 2, and 3, right? Yes. Out of 1, 2, and 3, who are the carriers? Who are the carriers? 2 and 3. So what is the carrier frequency? Can I write that the carrier frequency is 2 out of 3? Yes or no? Yes, because one is disease. Okay. So over here, if I tell you that since it's an autosomal recessive condition, if I tell you that if a person has cystic fibrosis, then it's 100% sure, we're 100% sure that both mother and the father were carriers, yes or no? Because because in order for the disease to happen, you need 2 recessive genes. So both of them have to be carriers. So for example, if this is the mom and this is the dad, then both of them, they're carriers, right? Okay, good. So from over here, we found out that the carrier frequency for the dad was, what was the carrier frequency? 0.006, right? Because this is what we figured out from the general population, if the disease frequency is 1 out of 90,000. If this is the carrier frequency for the father, what is the carrier frequency for the mother? Can we assume that the carrier frequency for the mother is 2 out of 3? Yes or no? Yes or no? Okay, so the carrier frequency for the mother is 2 out of 3. Okay, this is the chance of the mother being a carrier. This is the same chance as of the mother being a carrier. It's not the same as the father. Once again, it is not the same as the father because we calculated the father's chance of being a carrier because it was already said that the mother was a carrier. No. It was already mentioned that the mother was in the question. It said that the mother was the carrier, but we did not know that the father was the carrier too or not. The chances of the father being the carrier is this. Since the mother is already a carrier, which was mentioned in the question, the carrier frequency for an autosomal recessive disease is 2 out of 3. Are we clear about this? Yes or no? Are we clear about this? Once again, why carrier is 2 out of 3, not 2 out of 4? 2 out of 4 is not, it's not 2 out of 4 because one of them is already diseased. Carrier frequency is the frequency of diseased allele with, the carrier frequency is the frequency of the carrier alleles to the healthy alleles. Okay, carrier alleles to the healthy allele. That is 2 out of 3. Okay. Yes, I thought autosomal recessive is 1/4, carrier is 50% chance, having disease is 25% chance. Right, no, I understand that. But over there in the question, they mentioned that the mother is already a carrier. So the fact that the mom being a carrier over here, let's say, let me repeat the question once again, okay? Let me repeat the question once again. The question is, mom and dad, they want to know if their son has cystic fibrosis or not. Okay, are we clear about the question? Mom and dad, they want to know if the son has cystic fibrosis or not. Okay, the mom has a carrier frequency, or the mom is a known carrier of cystic fibrosis. If I tell you the mom is a known carrier for cystic fibrosis, what is the allelic frequency? The allelic frequency, not the chance, the frequency of the allele. Right. I understand it's 2PQ, but if they mention that the mom already has a carrier, or the mom already is carrying the gene for cystic fibrosis, then can we say that the mom is 2 out of 2/3? Because over here, 1 is the diseased, and then out of the 3, 2 are the carriers. So 2 out of 3 chance is mom is being the carrier. That's what we mean. Are we sure about this one? If not, then I'll repeat it again. Okay. No. Okay. Again, mom and dad, they want to know if the child has cystic fibrosis or not. The mom is a known carrier of cystic fibrosis. The dad's frequency is not known. What are the chances of the son having cystic fibrosis? This is the question. First of all, if the mom is a known carrier of cystic fibrosis, then the mom's allelic frequency is 2 out of 3. How did we come up with 2 out of 3? If we draw a genetic plot like this, one is the diseased, and both of them are carriers, and this is not 2 by 4, this is 2 by 3, because we're talking about carrier frequency. Carrier frequency is the number of carrier genes to the number of healthy alleles. So 2 out of 3, that is the carrier frequency. Okay, that is the carrier frequency. AA is also a carrier. AA is not a carrier because over A you have 2 dominant genes. It's an autosomal recessive condition. So 2 out of 3 meaning only the genes which contains A, the small letter A, this A. This A is the ones who are the carriers. Have you guys understood what I just said, or? But why then we assume that is AA if it's unknown if this in this way? We do not know if the dad is 2, or we do not know if the dad is 3, because the dad could be 1. We're not sure about this because the dad is not a known carrier of cystic fibrosis. The dad is not a known carrier of cystic fibrosis. The mom is a known carrier of cystic fibrosis. The mom is a known carrier of cystic fibrosis. If the mom is a known carrier of cystic fibrosis, the mom has an allele, which is AA. Okay? So the carrier frequency of AA is 2 out of 3. That is 2 AA out of 3 individuals. Are we clear about this? Okay, you guys are writing a lot of things that is really confusing me one by one. Okay, one by one. Who are the students who have not understood what I just said? Just say me. Okay, 1. Who else? Me. Okay, who else? Okay, okay, good. Let me get the first student and then one by one. Okay, okay. Let me get the first student. The first student was Dr. Karwasi. Okay, please unmute yourself and ask me what your confusion is.
Yes, good morning.
Yes, which
confusion? Because you mentioned she's a non-carrier, right? So then it means
I mentioned she is a carrier.
Oh, she's a carrier. Oh, okay, because I understood she. But yeah.
Okay, let me mention the question once again. The question is, mom and dad, they want to figure out if their child has cystic fibrosis or not. The mother is a known carrier of cystic fibrosis, and the dad's carrier status is unknown. What is the chance of the dad being a carrier if the disease frequency in the population is 1 out of 90,000? And if the dad is a carrier, what is the chance of the child having cystic fibrosis? It's a very complicated question, but this is the entire question. Okay, so having said that, let me break it down again. The question said, what is the possibility of the child having cystic fibrosis? So from our previous breakdown of 2PQ, we concluded that if the disease frequency in the population is 1 out of 90,000, the dad's carrier status is 0.006. Now, if the dad's carrier status is 0.006 and the mom's carrier status is already known, it's already known. If it's already known, then we can make an immediate assumption that the mom's carrier status is 2 out of 3. Why? Because out of this plot over here, number 4 represents the diseased allele. So this one is out. This one will not be considered. Now,
because she's a known carrier, right?
Right. She's a known carrier. If she's a known carrier, then she must be either 2 or she must be either 3. And the possibility of her being these 2 out of all 3 of them is the fraction of 2 by 3. So the mom's carrier status is 2 by 3. Now, have we understood till this point, Dr. Karbasi and everyone else?
So it means a non-carrier means she doesn't have the disease, but she can still be a carrier, right?
Of course,
so she
yes,
can be healthy and okay, I got my confusion. Sorry. Okay, I got it.
No need to apologize. You can ask me questions as many times as possible. Okay.
Thank you.
Doctor, wasn't the question we solved with the answer was about baby carrier chance? No, we are solving the question about the baby being diseased. We are solving the question about the baby being diseased. Have we understood this till now? How we came up with 2 by 3 and 0.006? Yes or no? Okay. Now, what is the next part of the question? The next part of the question is, what is the possibility of the baby having cystic fibrosis, right? Now, if I tell you that the baby has to have cystic fibrosis, what are the chances of the baby developing the diseased allele? Can we say that the chances of the baby developing the diseased allele is 1 by 4? 1 by 4? Yes or no? Yes? Because out of all 4 of them, yes, 1 by 4. So what is the possibility of the baby having the diseased allele? The possibility is mother, that is 2 by 3, into father, 0.006, into baby, 0.25. Would anyone be kind enough to do the calculation for this, please? What is the answer? 2 by 3 times 0.006, 0.25. What is the answer?
It's 0.001.
0.001. This is your answer. Okay. This is one of the most complicated questions you will receive in UWorld. And that's why I specified this question. If you can understand this question, you can understand all the questions of Hardy-Weinberg principle. Okay. You will receive this question in UWorld 100%. 100%. And the answer you're looking for is 0.001. I don't mean to give the answer out, okay? But I just want to make sure that you guys get the answer right, okay? Sorry for the cough. I think I might be coming up with a cold or something. Okay, but that's that. Are we clear about this? Hardy-Weinberg principle, very, very high yield. I have a question on this. Can we do it at the end of the lecture? Oh, okay, of course, we can do it at the end of the lectures. But that's that. Okay, if we have understood Hardy-Weinberg principle, first of all, can I ask you guys to write this down in your book, because you guys will forget about this. You guys will forget about this question. Do we have anyone who has written it down in their book, so that they know exactly how to study the Hardy-Weinberg? At least, if nothing, write down Q square equals to disease, P square equals to healthy, and 2PQ equals to carrier status. I'll write that down. Okay. Okay, my question is, when you construct the cross-linking square, are we doing mom versus dad or mom versus inheritance chance? Okay. Okay. First of all, the fact that the mom is a carrier status, the mom being a carrier, this is from the patient's, okay, who's the patient we're talking about? The mom or the baby? Who's the patient? The mom or the baby? Who's the patient? The baby. If I tell you that you have to find out the mom's carrier status, whose genetic table do you have to draw? The parents or the grandparents'? Whose genetic table do you have to draw? The parents' table or the grandparents' table? If you have to find out the carrier status of the mom. Fast answers, please. Grandparents, there we go. If you want to know the carrier status of the mom, you have to draw the table of the grandparents. So that must mean that if the mom is a carrier, then the grandparents should have been some sort of a carrier too. Yes or no? Except the fact that maybe none of them in the mom's generation was in that 1/4 to have the disease. They were all in the 2/3s. Yes? Okay. So this is the carrier status of the mom. That is the answer to the first question by Dr. Naud. The second question is, why did we multiply everything? By Dr. Ala. We multiplied everything because that's the procedure of coming up with the frequency. The frequency, if you want to calculate the frequency of an allele, you have to calculate the possibility of having the allele into the possibility of the mom being a carrier, and then into the possibility of the dad being a carrier. That's how you come across this one. Or else everyone would have had cystic fibrosis. Because if you say that 1/4 of an entire generation has cystic fibrosis, then you are saying that out of everyone in the world, 0.25 or 1/4 of the world has cystic fibrosis, and that is not the correct thing to say. You would have to say 1/4 of the world has cystic fibrosis if the mother has a carrier frequency of this number, and the father has a carrier frequency of this number, meaning that it should not be 0.006. It depends on the disease frequency in a population. Once again, it depends on the disease frequency in a population. Are we clear about this? Right, mom is already a carrier, yes. I forgot how we got 0.006. If the disease frequency Q square equals to 1/90,000, which was mentioned in the question, then Q equals to 1/300, and then P equals to 1 minus 1/300. That is 0. How much? 0.00009, I guess. So the answer is 2 into 0.9 into 1/300, and the answer is 0.006. That's how we got this one. In URL was the same question, but they said the mother has a probability of 50% because she's a carrier but healthy. Or we can talk about the question at the end of the lecture. Which question was that one? That said that the mother has a probability of 50% because she's a carrier. The mother has a probability of 50%. Okay, what is the real question in the URL? What is the question which you face? Because it must be a new question then. What is the question which you face in URL? Should I unmute? Yes.
Okay, like it was almost similar. It was like a healthy 30-year-old woman comes to the office as she and her husband desire a second child.
Okay.
The husband is infertile, and the patient's son, who was conceived via donor insemination, was recently diagnosed with glycogen storage disease type 2, POM disease. This rare autosomal recessive disease is known to affect 1 in 40,000 of the general population. Genetic testing confirms that the patient is a carrier for the disease. A different sperm donor is selected with no personal or family history of POM disease. However, his carrier status is unknown. What is the probability of the patient having an affected child with the new sperm donor? And they did almost the same what you mentioned us, but they say in the explanation, in this example, genetic analysis confirms that the mother is a carrier, which gives her a 50% chance of passing one mutant allele to her child. And they did the same equation, everything, but they use 50% and not
they use 1/2?
Yes.
Instead of 2/3?
Yes.
But was it mentioned that the mother had a 50% chance of being a carrier?
Like in the question was just written that she is healthy, but they had and the husband is infertile, and they got the child by donor insemination, and then was recently diagnosed with
what type of a disease is Pompe's disease? X-linked recessive or autosomal recessive?
It's written rare autosomal recessive disease.
Rare autosomal recessive, right?
Yes.
Okay, then if that's the thing, then let me get back to you on this tomorrow, because there must be confusion.
Okay, or if you want, Dr. Hatari, I can also screenshot it and then send it to the group if you want, and with the explanation.
Yes, you can do that too.
No offline. Okay, I will do that.
Okay, you will do that too. But having said that, if we can come up with the solution for 2 by 3 or 1 by 2, have we understood at least the Hardy 1-bar principle?
Yes.
How we are trying to come up with these things?
Yes.
Okay. Okay. Just give me 1 second. Let me come up with my question so that one second. Let me come up with my question. Okay. Let me come up with my question. Let me write down the question which I got from UWorld a long time back. Okay. Please write this question down. Okay. Fortunately, I have a question. I have a solution for all the confusion right now because I made a small mistake. Okay. This is the question. A couple wants to know if their child has cystic fibrosis, the carrier frequency of the father is unknown and the mother's frequency is 2 by 3. They mentioned the frequency in the question. Okay, they mentioned the frequency in the question. Now, is it better? Okay. Okay. Okay, my apologies. I was just a little bit confused about this one over here. Okay. Okay. So the carrier frequency is 1 by 2. Okay, my apologies. Because I got confused with the question. Okay, so you guys were correct. I was wrong. I told you guys the carrier frequency is 2 by 3, which is not the right thing to say. The right thing is carrier frequency is 1 by 2. Disease frequency is 1 by 4. Healthy frequency is 1 by 4. Is everything clear now? Okay. Okay, the question mentioned that the mother had a carrier frequency of 2 by 3. Now, everything makes sense. Okay, I had to go through the question once again to come up with what was going on, because most of the time I make sure that I remember everything before I produce the lectures, but at times I get a bit confused because I have so much on my mind. So that, okay. Yeah, I got a question, they say 2 by 3. Okay, so that's that. So they tell you that the mother's frequency is 2 by 3. So that's the whole thing. Is everything clear now? Can you repeat the frequency again? The frequency is 2 by 3 into 0.006 into 1 by 4. Okay. So with that being said, did we understand the Hardy-Weinberg principle? If I have to ask you to calculate the carrier frequency, which equation will you use for the carrier frequency? Very good. How about the disease frequency? Okay. How about the healthy frequency? What is the total amount with healthy and diseased? One. Okay, good. Can you repeat the clarification? The clarification was the question mentioned that the mother is 2 by 3 carrier, 2/3 carrier. Okay, so that's why the amount is 2 by 3 into 0.006 into 1 by 4. Okay, are we clear about this now? And if the question does not mention, then we can say that the mother is 1/2. Yes. In the normally carrier frequency is 1/2. Can we do another question on this? Unfortunately, I do not have another question on this. This is the only question which I thought of. I actually had this question in my mind. That's why I had to come up with, that's why I made the mistake which I did, because I did not read this. So once I read it again, I realized that the frequency was mentioned in the question. That is 2/3. So that's that. You have a question, sure. Let's do your question. Can we do another question on this? Okay. Okay, let's see what your question is. A healthy 20-year-old comes to the physician with his wife for counseling. His sister died of cystinosis and autosomal recessive. The incidence of this disease in the population is 1/40,000. The wife's history is non-contributory. The wife risks for being a carrier of this is closest to which of the following? Okay, so let's do this. Should be pretty easy. What is the risk of the wife being a carrier? First of all, if the disease frequency is 1/40,000, that is Q squared, then what is Q? What is Q? 1 by? Very good. If that is Q, then what is P? Calculate for P. Then calculate for the carrier frequency. Calculate for the carrier frequency and see which one is closest to which number over here. Which one is the closest to which number? Out of all the options provided by Dr. Tareem, which one is closest? D. Okay. Is D the correct answer? Is D the correct answer? Dr. Tareem? Because with the world, you never know. There could be another. It's B. What mistakes did we make? Let's see. Let's go back to this one. The wife's risk for being a carrier of this disorder is closest to which of the following? Okay. Would you be kind enough to provide the explanation for this in the discussion group? Actually, it's D. Okay. Okay, good. So you had me a bit, you scared me. Okay, good. Because the last thing I want to be is look bad in front of you guys. Okay, because I don't want to give you guys an explanation and then come across looking stupid. Okay, so I'm glad it worked out for everyone. I'm pretty sure some of you guys were like more like, this guy who was teaching us is complete bullshit. He knows nothing exactly of what he's talking about. Okay, but thank you so much for saying that it's D. Okay. Okay, good. So thank you so much, Dr. Tareem, for sharing those questions. And Dr. Karbasi was also kind enough to share the questions about Hardy-Weinberg principle in the discussion group. And you guys can also go through the discussion group to go to some of the questions which he has provided. And that would also be a good source of revision. Okay, so having said that, have we finally understood how to target the Hardy-Weinberg genetic questions? Yes or no? Okay, good. So with that being said, can we study the uniparental disomy and imprinting disorders tomorrow? Because if we start talking about Prader-Willi and Angelman syndrome, it will actually take us some time. So we wanted to jump into some UWorld questions. Okay. We wanted to jump into some UWorld questions and discussions. Can I ask one more thing? Yes, of course you can ask one more thing. If you need to unmute yourself, you can. If you want to use the chat box, you can, no problem.
And one question, like about the question which I posted.
Yes.
If they would not know that the first child has Pompe disease, but they generally want to know that is there a possibility that the child can get Pompe disease, then I think then the mother has to be 2 out of 3, right? Because she can be completely healthy, but she can also be a carrier. If like
right. Then that's the issue. That's where the confusion is. So what you just mentioned was, if they tell you that the first child did not have the disease, then the possibility of the mother being a carrier is actually 2 out of 3. Because what I was thinking was, if you have, let me take another blank page. Okay. If you have a patient, okay, generally in a population of autosomal recessives, okay, in a populations of autosomal recessives, okay, generally you have these 2 who are the carriers, and out of general population, if there are diseased and healthy individuals, then out of diseased and healthy individuals, the population of carriers is 1/2. Okay, but if they tell you that in the populations who are not diseased, okay, for example, out of populations who are not diseased, if they ask you directly what is the carrier frequency out of all the healthy carriers, what is the frequency of carrier out of healthy carriers, then the answer is 2/3. Okay, are we clear about this? There are 2 ways you can be asked. The question can be asked in 2 ways. The question can be asked 2 ways. Okay. Is everyone clear now? Are we talking about different things over here? Are you guys talking about something which I'm not talking of? Which I'm not talking about? Yes or no? Okay. What are you guys talking about? Okay. Okay. What I was saying was, over here, if they ask you what is the carrier frequency out of healthy and diseased, the carrier frequency is 1/2. If they ask you what is the carrier frequency out of only the healthy, the frequency is 2/3. Okay. Are we clear? Okay. So with that being said, can we stop with the first aid reading right now and move on to questions or notes? Okay. Can we take a small 5-minute break before we jump into the questions? Perfect. Okay, thank you everyone for answering Dr. Allah's question, which I could not answer myself because I didn't even realize what she was asking. Let me go see what Dr. Allah was asking. Okay. You guys have been discussing for quite a long time while I was talking about something else. Okay. I have a silly question, maybe because of tiredness, I don't know, but how is 0.009 close to 1/100? Did you get your question? Did you get your question answered properly, Dr. Allah? If not, then I can. Okay. Okay, you are welcome. Now the question by Dr. Salim, is it safe to say for someone who has family history, we use the 2/2 table, and for unknowns with no history, we use Hardy-Weinberg? Yes. With no history, general population and then disease frequency, you can use Hardy-Weinberg. Okay, but if you have to come up with the final answer, then you have to draw the 2x2 table, or else you wouldn't know for the final answer. Okay. Okay, so with that being said, thank you so much for understanding the Hardy-Weinberg's principle with absolute perfections. Thank you to all the students who has contributed with questions to help us out even more. So with that being said, what we would do is we would take a 5-minute break, and then after that we would come back and do the UWorld questions. Okay, so let's take a 5-minute break right now. Okay, is everyone back from the break? Is everyone back from their break? Okay, good. Can we begin with the usual questions? Okay, good. Thank you so much. Thank you so much, Dr. Tharin, for sharing the videos on Hardy-Weinberg equations on YouTube. If you guys get the chance, please go through the videos, and that might clear up any confusion which you might have. But having said that, have we understood, once again, I'm sorry for asking the same questions over and over again, but have we understood the Hardy-Weinberg's principle, yes or no, about how to target the answers? 2pq, p square, q square, p plus q equals 1, and everything else? Okay, good. So without further ado, let me share the screen. Let me get done with the questions. Okay. Okay. So can you guys see this question over here? Can you guys see my screen properly? Can you guys see the question? Okay, good. So let's read this question. Which of the following mechanisms is the most likely cause of this infant's clinical findings? Once again, you have only 90 seconds to solve the questions. A 1-hour girl born to a 40-year-old woman is brought to the nursery for evaluation. The pregnancy and delivery were uncomplicated. Physical examination shows mid-phase hypoplasia with a flat nasal bridge, slant and palpebral fissure, small mouth, and a single palmar crease. If you guys are ready, please let me know you're ready, and let's begin. Okay.
I'm ready.
Okay, good. What is the answer?
B,
Please.
mosaicism.
Mosaic. Okay, good. Everyone in the chat box, please. Okay. I think it will be better if we all use the chat box. That way every one of us can participate. So if you have the answer ready, please write the answer down in the chat box. And B is for mosaicism. Okay, mosaicism is Down syndrome, exactly what we just talked about. Can you guys tell me another example of mosaicism? What is another example of mosaicism? McCune-Albright syndrome. McCune-Albright syndrome. Very good. Okay, next one. Okay, 1 second. Okay. Okay, next one. Okay. Which of the following enzymes is most likely to be deficient in the newborn? A 23-year-old woman gives birth to a full-term neonate. The newborn's vital signs are within normal limits. Physical examination shows ambiguous genitalia and clitoromegaly. The laboratory studies reveal elevated serum levels of testosterone and androstenedione. The karyotype shows 46XX. Ultrasound of the infant's pelvis shows a normal-sized uterus. The mother experienced facial hair growth and voice deepening during the pregnancy. Very easy to understand that one enzyme is not working over here. As a result, the mother has increased action of one type of enzyme or one type of hormone. If everyone is ready, please say that you're ready and mention the answers in the chat box. What is the most likely answer? What is the most likely answer? D. D, aromatase. Aromatase is responsible for converting testosterone to estrogen, so aromatase deficiency. Are we clear about this? Yes or no? Okay, next one. Which of the following is the most likely cause of this patient's condition? A mother brings her 1-month-old son to the ER because he appears floppy and has not been feeding well. The mother is a 43-year-old multipara, and the family recently emigrated from Eastern Europe. Physical examination reveals mild jaundice, enlarged tongue, and general hypotonia and an umbilical hernia. This is a pretty complicated question, so please take your time. And if you're ready, tell me the right answer. Anyone? What is the most correct answer? Very good. What is the diagnosis for this patient? Cretinism. Very good. Cretinism. Okay, cretinism. Okay. Okay, 1 second. Okay, let's do this. A 24-year-old African-American woman comes to the office with her husband for a prenatal counseling. She has a 3-year-old child with sickle cell anemia from a previous marriage, and the child's father died in a car accident. The patient remarried last year and is interested in having more children. She and her new husband do not have sickle cell anemia, and the patient's husband has no other children. However, the patient and her husband are worried that the future children could have sickle cell anemia. A urine pregnancy test is negative. Which of the following is the best test that can be offered to this couple? Which one is the best test that can be offered to this couple? If you guys are ready, let me know you're ready. Okay, let's give everyone some more time. Okay, what is the right answer? Okay. First of all, we have to do a hemoglobin electrophoresis to identify sickle cell disease. Why do we do the hemoglobin electrophoresis for the mom and for the dad? Does anyone have any idea? The maternal and the paternal hemoglobin electrophoresis. First of all, the correct answer is D. Okay, the correct answer is D. D is D. D for Danny. Danny. Okay, D is paternal hemoglobin electrophoresis. This is the correct answer. Okay. Have you guys understood? Are we okay? Are we clear about this? Okay, because in sickle cell diseases, what happens is that the mother is the carrier, right? What happens is that females are carriers and males are sufferers. That's why we do the. Do you remember that one? That females are carriers and fathers or males are the sufferers. That's why you do the. That's why you do the paternal hemoglobin electrophoresis. Okay. Yes. Okay, last question. Last question. Not clear. Okay, what type of a disease is sickle cell anemia? What type of a disease is sickle cell anemia? X-linked recessive. X-linked recessive. How many X chromosomes does the mother have? 2. How many X chromosomes does the father have? So if it's a recessive disease, if the father has only 1 X chromosome which can be damaged, then will the disease be more likely in the father or in the mother? Will the disease be more likely in the father or in the mother? It will be more likely in the father. If it's more likely in the father and we have to do hemoglobin electrophoresis, why do we can just focus on the hemoglobin electrophoresis of the father instead of the mother? Am I correct? Yes or no? Or if we do it for the mother, right, it's easier to detect. If you do the maternal hemoglobin electrophoresis, you can also get sickle cell diseases, but if you do the father's one, it's confirmatory. Now, are we clear? Are we clear now?
Doctor,
Yes.
my question is because if you are going to the concern is about the future offspring,
Mm-hmm.
and
Yes.
the mother can be a carrier because
Right.
she had a previous defective or a child with sickle cell.
Right.
Therefore, the main concern is whether the mother is a carrier, because the father being a carrier is very less likely. If he has the disease, if he is a carrier, he cannot be a carrier, because if it will be manifested as a disease, I mean.
Okay. You know what? Let's read the question again. I'll break it down again. Okay. I'll break it down for you. Let me read the question. Okay. A 24-year-old African-American woman comes to the office with her husband for prenatal counseling. She has a 3-year-old child with sickle cell anemia from a previous marriage, and the child's father died in a car accident. The patient remarried last year and is interested in having more children. So if she wants to have future children, now if she wants to have future children and she wants to find out if her future child will have sickle cell anemia or not, then she already knows that she's a carrier, so she can easily do so there's no need to do the hemoglobin electrophoresis for the mother, right? So if she has to do hemoglobin electrophoresis, she has to do the hemoglobin electrophoresis for the male that she will be with in order to diagnose whether her future children will have sickle cell anemia or not.
I think we made a mistake because sickle cell is autosomal recessive, not X-linked.
Sickle cell?
Autosomal recessive.
Give me 1 second, please. Just give me 1 second. Hello? Okay, so where were we? Okay.
Yeah.
Okay. Okay, so once again, is sickle cell disease autosomal or X-linked? Which one?
It autosomal recessive, and we sure the mother is carrier. We have to do it for the father.
Exactly.
Because the patient need 2 genes to be affected.
Okay, so let's go back to the question once again. This question is not about the fact whether the disease is autosomal recessive or X-linked recessive or not. But over here, who was the doctor who just helped me out with this question?
Iman.
Okay, Dr. Iman, thank you so much. What she mentioned was that it's over here, it's already mentioned that the mother is a carrier. And all we have to do is you have to figure out whether the father is a carrier or not. That's the only reason why you have to do the paternal hemoglobin electrophoresis. Because if the mother previously was a carrier, and that's why the child had the sickle cell diseases, then if she wants to prevent a future pregnancy with sickle cell disease, then she has to make sure that the person she is going to be with, he does not have sickle cell anemia. That's why the paternal hemoglobin electrophoresis is the right answer. Are we clear now?
Sorry for that stupid question, but I got confused because I thought it's C because it's a mismatch disease. So an orphan blood is using for RNA. So could I not use it, or do I'm confusing everything?
You could use it but it's not the initial test. You understand what I mean? Over here it says which of the following is the best initial test. Okay. You can probably do this later, no problem. Okay.
And paternal karyotype would not give me, if it's sickle disease, it would just show me the chromosome abnormalities, right?
The hemoglobin electrophoresis?
Yeah, paternal karyotype, analyzing what's also written.
The paternal karyotype. If you do sickle cell diseases, no. If you do a fish, then most probably you could see some sort of abnormalities, but karyotyping, no. If you do any karyotyping, you won't see any abnormalities.
So I would just see chromosome abnormalities, but not specialty disease. Okay.
Right.
Okay, got it. Thank you.
You are welcome. Okay, can we move on to the last question?
Yes.
This one, please. Okay, let's look at the last question. A 32-year-old male presents to the ER with recent onset of fatigue, dyspnea, and fever. Cytogenetic studies of this patient's blood cells demonstrate a 15-17 translocation. Which of the following protein is most likely malfunctioning over here? Very easy question. We have talked about this in the past a lot of times. And if anyone is ready, please say you're ready and let's do this. Very easy question. Everyone else? Okay. What is the right answer? Retinoic acid receptor. Retinoic acid receptor. Okay, very good. Do we have any confusion with this question? Okay, good. Very good. So with that being said, thank you so much for doing the questions. Okay. And with that, I would like to conclude the lecture for today. And I would like to discuss the UWorld notes tomorrow. But we do have a short, we were told that we have a lot of students over here who is willing to take the exam in 2 to 3 months. So I wanted to talk to those students to see whether they were ready and what we could do to potentiate their timing so that they can do well. So if you are one of those students, please stay behind. If you are a student who is willing to give the exam in 2 to 3 months, please stay behind and we will talk about our discussion in 5 minutes. If you are not going to give your exam in 2 to 3 months, then thank you so much for doing the lectures. I'll see you guys tomorrow at 9 a.m. If you guys have any questions, please don't hesitate to send us an email. And I hope you guys have understood today's lecture. Once again, for everyone who needed an extension, please make sure to complete the transactions by tomorrow. And if not, please let us know if you have any issues. We'll try to work around it. And try to do the UWorld question, guys. Okay, do the UWorld questions. If you don't do the UWorld questions, you can say bye-bye to your genetic knowledge. They will fly away faster than the fastest bird in the world. Okay, so that's that. And so with that being said, okay, we have another question. Are endocrine lectures available? The endocrine lectures, we are not available, Dr. Salim. I apologize, but we will try to see what we have. But you have access to our initial lectures. Over there, we have some endocrine lectures from lecture number 1. If you try to see lecture number 2, 3, 4, they should be about endocrine. So please try to see if you have access to those lectures. If not, let us know. We will try to see if we can give access to you. Any more question? Do we have any more question? Do we have any more questions from anyone? Okay, if no one has any questions, then for all the students who are willing to give the exam in 2 to 3 months, stay behind. We will start the discussion in 5 minutes. And for everyone else, you guys are welcome to say goodbye, and I'll see you guys tomorrow. Okay, thank you so much. Have a great day. See you guys tomorrow. Don't forget to do the UL questions. For everyone else, we'll give the exam in 2 to 3 months. Stay behind. I will start the discussion in 5 minutes. Okay, let's take a break for 5 minutes. Let's come back so that we can start the discussion. For anyone else who wants to stay behind and hear the discussion, you are more than welcome to do so. No problem. Okay, but just give me 5 more minutes and I'll come back. Okay, is everyone here who wants to discuss about their preparations? Okay, so let's start with the discussion. Okay, so I know there's a lot of you over here who wants to discuss about this. Okay, so let me just give a very brief introduction to what I want to discuss about. Okay, just give me 2 minutes. I mean 2 seconds. 1 second. Okay. Okay. So we will begin with the discussion of the exam, that is the USMLE Step 1. First and foremost, the USMLE Step 1 is one of the most important exams you will give in your lifetime as a medical student, and also one of the most important exams you will give in your lifetime as an IMG, International Medical Graduates, because you are going to give this exam to get a score so that you can get into a proper residency program into a well-known hospital, which can help you out with your visas if you have any problems, especially if you're not a citizen or if you're not a permanent resident or a green card holder. So what type of scores are we talking about? Okay, what type of scores are we talking about? We are talking about scores above a minimum of 230 or more than 230. Okay, can you guys hear my voice? Can you guys hear my voice? Are you guys with me? Can you guys hear my voice? Okay, good. So we are talking about a minimum score of 230 or more than 230. Okay, the passing score of USMLE Step 1 is 196. Even if you give the exam in 2021, you will still end up with a score. What will happen in 2022 is far from our discussion, but if you give the exam in 2021, it's still a score-based exam. So basically, you will be judged on your score, especially if you are not a citizen or a green card holder, then you need to have a higher score on Step 1 minimum. Okay, having said that, everyone needs to have a good score. That's a given. But with scores with 220, especially for students who are green card holders, who are citizens, then we have citizens of Canada and everyone else, the minimum score for you guys should be 220. Okay, with that being said, do not listen to what people tell you. If you get a score close to 220, and most of the times everyone who gives the exam, none of them are bad students, okay, but there could be certain circumstances where you can end up with a score like this and you have absolutely nothing to be ashamed of or nothing to be worried about, because this is actually a pretty good score. I'm going to tell you why. Because with this score, you can match into medicine, family medicine, internal medicine, general surgery, and regardless of what people say that you need a very good score and this and that, a lot of it actually depends on your clinical experience, on your ability to give proper interviews, and then your research studies, and you always have your Step 2 CK to cover up. So with that being said, do not put a lot of pressure on yourself, because at the end of the day, you guys are just human and every one of you are doing the best you can. But this is the minimum score which we should be targeting. This should be a target score. Okay. Next point, which I want to talk about, is how do you know you are ready? How do you know you are ready to sit for the USMLE Step 1 exam? Basically, how do you know you can enter the dedicated period? The dedicated period. The dedicated period, once again, is a period of 4 to 6 weeks of intense studying of 10 or minimum 8 hours to a maximum of 10 to 12 hours, where you do the UL question, first aid reviews, pathomas. Basically, you try to stick to as less resources as possible and try to maximize that point. How do you know you are ready to enter the dedicated period? If your UL's overall percentage if your UWorld overall percentage is more than 85%. And in each and every block, you are getting more than 90% on a second pass. Not a first pass, on the second pass. The second pass, it should be more than 90%. That's one. Number two is NBME scores. If your NBME scores are above 210 minimum, this is a minimum. NBME score should be at least above 210. Okay, at least above 210 for you to understand that you can enter a dedicated period, because it could be NBME scores are always 210 plus minus 10. So 210 could actually mean you can get 220, or this can actually mean you can get 200, or even on good days you can get 230, or even on the worst days you can actually get 192. This is a very good score. This is a very bad score, meaning that you failed. So minimum of 210 is important to understand you are ready to enter the dedicated period. Number two, if you enter the dedicated period, then what do you do? Okay. What do you do when you enter the dedicated period? Now, this is the point where I want to talk to you guys about how to maximize your studying in dedicated period. Okay. First of all, I will tell you what I used to do when I was a student of step one. And later I can tell you what I did not do, which I could have done. First of all, what did I do? I would wake up early. Dedicated period, students for dedicated period. Students will give the exam in 2 to 3 months. Wake up early, or at least make sure that whenever you wake up, okay, by early, I don't mean wake up at 4 a.m. and start your work, okay? You can wake up at a minimum of 8 a.m. in the morning, that's okay. Or even if you wake up at 10 p.m., at 10 a.m., my apologies. If you wake up at 10 a.m. and you get to work, make sure that you at least get a sleep of at least 6 hours every day. 6 hours of sleep every day, minimum 6 hours to a maximum of not more than 8 hours. Maximum of not more than 8 hours, because every hour in your life of dedicated period is important, because throughout your whole dedicated period, you not only do you have to study, you have to cook for yourself, clean up, do chores around the house, get a workout in, have some time for some amount of socializations or relaxations, right? So out of that 24 hours, 6 to 8 hours should be done on sleeping, okay? So out of those 24 hours, 6 hours is gone. Now you have 18 hours. Out of 18 hours, you should study for a minimum of 8 to 10 hours. 8 to 10 hours. So let's say that you study for another 10 hours, right? Let's say 10 hours. Now you have 8 hours left. This 8 hours is your time for cooking, cleaning, socializing, working out, doing whatever you want to do. This is your time span. And another thing which I want to tell you guys is consistency is the key, basically meaning that if every day if you can study 8 to 10 hours, instead, if you study, let's say 15 hours in one day, and in the second day you study for 3 hours, okay, this is not good. This is not the right way to go. Every day, try to choose an hour which you can stick by consistently. 8 to 10 hours is pretty basic. Everyone should be able to do 8 to 10 hours minimum, because I know it will still be a little bit difficult for everyone to put in that amount of time. But since it's your step one exam, I would suggest nothing more than 8, and more than 10 is welcome, but nothing more than 12 hours per day. Nothing more than 12 hours per day, because you will go into a crash mode. Okay, so this is what this is what I would do. I would wake up early. Okay, I know a lot of our friends, they would wake up a bit later, but they would stick to their schedule no matter what. So consistency is the key. Always remember that. So I would wake up early. After I woke up, the first thing I would do is get into the zone of studying. How do you get into the zone of studying? So what I would do is I would have a timer. If you go to Google and you press timer, then you can get timers over there. So my first hour, I would time my hours, meaning what would I do? When I would study, I would go to Google. Okay, let me show you. Okay, let me show you what I would do. I would go to Google, I'd write timer, and I'd start like this. Stopwatch, start. Okay, and this would start, and I would start my first work. Okay, and let's say during this 1 hour, if I get a phone call, I would stop my time. I would receive that phone call, talk about like whatever I have to do, then hang up, and then start my timer once again. That's what I would do until and unless it's exactly 1 hour of studying, no matter what. Even if I have to go to the bathroom, I will stop the timer, go to the bathroom, come back, start the timer. And when this timer goes 1 hour, then I would be done with my 1 hour. That's how perfect my 1 hour is. And that's how perfect your 1 hour should be, that it's in 1 hour of absolute study. So to get inside the zone, the first thing that I would do is 1 hour, I would watch videos, any videos, get my mind used to think. The most important videos which I would choose to watch early in the morning to get into the zone and get my brains functioning is Pathoma, because everything is very concised over there. I would target, let's say, an hour of Pathoma every day in the morning. So once I was done with my 1 hour of Pathoma, okay, I would take a break of no more than 5 minutes. After my 5 minutes, I would use the next 3 hours to do UWorld blocks, random and timed. Are you guys still with me? Are you guys understanding what I am saying over here? Are you guys still with me? Or am I just talking to myself? Okay, okay, good. So first hour I would do Pathoma, get into the zone, get my mind ready, get ready for the questions I will get every day. It was like a war, me versus UWorld, okay? So 3 hours of UWorld blocks, okay? 3 hours of UWorld blocks. After that, I would take, so let's say if I woke up at 8 and it's 9 and then 10, 11, 12, and let's say 12:30, I would have lunch. Okay, I would have lunch, and then along with my lunch, after that, I would have a 10 to 15 minutes nap. That's what I would do. I would nap because I would want to get rid of the afternoon laziness, right? I would just get my nap out of the way. Then the next thing I would start with is a big glass of dark coffee, and I would start with my afternoon study period, which is first aid. This is how I would divide my first aid doing my reviews, because I realized the more first aid you study or the more first aid you do, it allows you to answer more questions perfectly. This is what I would do. First hour of first aid, 1 hour, I would focus on doing nothing but drugs, because drugs are one thing which I would do every day because they are very easy to forget. So I would do drugs every day. Let's say I would try to focus on finishing the pharma every week. Every week I would finish the pharmacology at least once. Especially I would target the pharma of the organ systems, the pharmacology portions of the organ systems. So every day I would read 5 to 10 pages of pharmacology. 5 to 10 pages of pharmacology every day. I would start with CVS, and by the end of the week, I would be done with reproductive and immunosuppressions and oncological pharmacology from the basic sciences. And I would do that every day. I would target 5 to 10 pages, and I would read them. I would test myself. I would underline, I would draw, I would do whatever. But 1 hour, I would talk about or think about pharma. Next 1 hour, exactly by my timer. Exactly by my timer, I would do pharma. Next 1 hour, I would do the bugs, micro. Every day I would do micro. Every day I would do pharma, no matter what. No matter what, I would do pharma and micro every day, because these are the parts of step 1 where the questions can be really easy or the questions can be really difficult based on your preparation. Because it's very easy to understand the pathologies, because you can associate a lot of questions when you think about pathologies. But if you do not have the information of micro and pharma in your mind, there is no way you can come up with the properties of a certain bacteria, for example, or the properties of a certain virus, for example. So I would make sure that my micro and my pharma was on spot every day. So once again, 5 to 10 pages. And every week I would be done, or every 1 to 1.5 weeks, I would be done reading pharma and micro at least once. So if I had 4 to 6 weeks of prep, I would make sure that I would read pharma and micro from first aid, especially 4 to 6 times. Okay, 4 to 6 times. Next 1 hour. Next 1 hour, I would read first aid by itself. What does that mean? For example, I would read CVS. I would always begin with the organ systems, and I would always go from the organ system. So let's say I would start with CVS, and I would read 5 to 10 pages of CVS every day. And then eventually when I would go to endocrine, I would go to endocrine, and then so far and so forth, then GI every day. Skipping out the pharmas and micro, because I am studying the pharmas on my pharma timing. So everything except pharma, I would read them from the organ system every day. 1 hour of organ system, 1 hour of basic science. Basic science, 1 hour organ system, 1 hour basic science every day. What does that mean? I would study biochem every day, 5 to 10 pages, more or less, until and unless I finished biochemistry. Then obviously, since I'm doing microbiology, so I would skip out on microbiology. After I was done with biochemistry, I would study immunology, 5 to 10 pages every day. Not on the same day. Keep in mind, these are not on the same day. Eventually, I would go from one to another. But every day in my afternoon session, I would have 4 hours of first aid divided systematically into pharma 1, micro 1, organ systems 1, and basic science 1. After that, by the end of the day, I would be so confident. I would that what I would do is I would take another break in the evening. So let's say it was 12:30, right? I had my lunch for 10 to 15 minutes. Then I started this session from 2 p.m. and I ended at 6 p.m., right? At 6 p.m., I would get a workout in. So I would either I usually work out with my wife. So me and the wife, we would have gone and hit the gym or something, or work out in the house. You guys can do whatever you want to do. Either go for a run. I would advise you guys to step outside of the house, stretch, do whatever you have to do. I would take a break from 6 to, let's say I would take a break to 8 p.m. 6 to 8, right? 6 to 8, I would take a break, do whatever I have to do in that time period, workout, shower, and then out of the 3 blocks over here, 3 URL blocks, I would focus on I would study for 1 more hour before dinner. I would solve as many of the wrong questions only wrong questions. Then I would have my dinner and another 1 hour, right? And after that, I would have another 1 hour of wrong questions again. Okay, so from 6 to 8, I would do whatever I have to do. I would work out. You guys can do whatever you want to do. Then before dinner, like let's say I would usually take a shower. Then after shower, I would sit down with my 1 hour of wrong questions. Then I would have dinner, watch a series, and before going to bed, I would solve another 1 hour of wrong questions. Wrong questions of that single day, And this is exactly what I would do for the next 4 to 6 weeks, each and every day, consistently. How many times a day or how many times a week would I do this? I would do this 6 days per week. Consistently, I would do this 6 days per week. So you over here, you have 4 hours, then you have 4 hours, then you have, that's 8, 9, 10, 11. This is how much I would study every day, broken down systematically one by one, one by one. I would make sure that my timer goes off and I would click, 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 click. And then what I would have is I would have this sort of routine. Okay, let me show you the routine I would have. Okay, so what I would do is I would start my day. Before I start my day, I would have a piece of paper where I would write 1 hour pathoma, click. 3 URL blocks, click, click, click. Then first aid pharma, click. Micro, click. Organ system, click. Basic science, click. Wrong questions, click. Wrong questions, click. And then until and unless every one of them was clicked out, and I would personally make sure that I clicked, because when I clicked or when I put the right across my task, then this would give me a satisfaction that I have done it completely, So that's what I would do every day. And I would make sure to click it. And I would not go to sleep until and unless every one of them is clicked out. You understand what I mean? Although you would obviously have days where you would not want to study as much. Okay. So, but still, I would make sure that you guys do as much as possible. So this is what I did. Okay. What I did not do. Okay. This is what I did. What I did not do was I did not do this. Anki. I did not do this. Okay, I did not do that. Instead of doing Anki, I would read first aid every day. And by the end of my dedicated period, if I closed my eyes, then I could read first aid in my sleep. That's how much of first aid I have read in my life. Every day I would read first aid for 4 hours, and by the end of the last week of my exam, I remember I would lie down and I would close my eyes and I would read first aid and I would not basically sleep as per se. What I would do is I would close my eyes and then I would try to remember what was happening in the chapter of genetics and this and that. And I would try to remember if I remembered everything. If I could not, I would rush to the book, see what it was, and then go back to sleep again and just try to see what was what, because I was that obsessed with perfecting first aid. So that's the best advice I can give you, because if you have first aid in your mind, then everything in USMLE will be very easy for you. What I did not do was Anki, and I highly suggest if you guys have the possibility, instead, let's say you guys do not wish to read first aid as obsessively as I do, then the alternative for this one is Anki. That's what you guys can do. But with that being said, this is the best advice and this is the best routine schedule I can give you right now. So for all the students who are willing to give the exam in 2 to 3 months, my advice to you is, first of all, you have to assess for yourself whether you are ready to enter the dedicated period or not. If your UWorld score and NBME scores are good enough, then I would suggest do not waste any time, enter the dedicated period and lead this kind of a lifestyle for 4 to 6 weeks and you guys will do amazing regardless. If your UWorld and NBME scores are not up to the mark, let's say your NBME scores are close to, your UWorld score is 75% on second pass, NBME scores are 200 or 195 or 198, I highly suggest you guys take some more time, read first aid once again, and postpone your exam. Okay, postpone your exam. That is my best advice to you. And with that being said, I am done. Now let's hear it from you guys. What questions do you guys have for me so that I can help you guys out? One by one. Anyone? What questions do you guys have? Have you guys understood the routine? Okay. Does anyone have any questions? Does anyone have? Okay. Please introduce yourself, Dr.
Dr. Charlene.
Yes, hi, how are you? What is your question, please?
I am good. How are you? My question is, when you're reviewing your UWorld questions, how, like, when you say review, like, are you just reading like the educational objective or really like getting to know what you got wrong?
Okay, good. So, so the question asked was how to review the UWorld question perfectly. Okay, so if I'm telling you that I'm reviewing the question, that means that if I'm doing the UWorld questions once again, this is obviously the second or third pass of my UWorld. Okay, so by the second time or the third time that I'm doing my UWorld, and if I'm making the same mistakes again and again, Okay, then what I would do is by reviewing, first of all, I would review. Okay, so if these are my answer options. Okay, first of all, if I answered B, but B is not the right answer, and let's say D is the right answer. Okay, first of all, I would like to correct myself. I would like to see why I thought B was the right answer. Before seeing why D is the right answer, the first thing I would do is read the question once again, see what I missed out. Then I would correct myself because I have to change my way of thoughts. You have, I mean, me as a student, if I have to give the exam again, then I have to change the way I'm thinking. So the best way I have to do this is, first of all, I have to get rid of this habit of making the wrong choices by seeing why I made this wrong choice first. So first of all, I would see why B was the wrong option. Then the next thing I would see is D, is why is D the right option? Okay. So after I've done that, basically, I have basically understood what's happening, and I'm done with 95% of my review. The rest of the 5% is I would quickly glance through A, C, and E to see what they're talking about. If I know the information in A, C, and E are the informations I've seen before, or if there's any new information over here, then I would quickly go through them. I would not really go through the educational objectives as much because because I have been reading first aid so much that I know exactly why I made the wrong answer. But with that being said, I would go through them. I would just roll my eyes throughout the objective, not really put a lot of attention to it, because I'm pretty sure I have put my attention to it in the first pass when I was doing them very carefully. So since this is the pass of UWorld I'm doing in my dedicated period, once again, the first thing I would do is read the wrong answer, why I made the wrong choice, correct myself, change my thought patterns, then understand why D is the correct answer, quickly glance through ACE to see if I have all the information over here, and then just glance through the educational objectives, or at times I would even skip out on this one. I would not even care. That's what I would do every time, because if, let's say, 3 blocks of questions is 120 questions. Out of 120 questions, if I made 40 wrong choices, then it would take how long? Let's say I would take nothing more than 2 to 5 minutes maximum per review. 2 to maximum 5 minutes maximum per review, because I would not waste a lot of time reviewing the answer, because my goal was that I have to get everything ticked out properly so that tomorrow morning I'm not late and I'm not missed out. Because in my dedicated period, my goal was to focus a lot on quantity of questions as well as quality. But since I have focused on the quality of doing questions in the first pass of UWorld, I would equally focus more on my quantity of questions. So that's the thing. That's what I would do. Any more questions?
I have a question. It's
Okay,
Jordan.
Yes.
So you told us that we can depend on UWorld whether we should go to dedicated period or not. But I honestly feel like, don't you think like when we do the questions for the second time, it's more of like memorization of the questions rather than the concepts. Okay. So don't you think it will over predict our results?
Okay, good. Very, very good question. Okay, so first of all, the question that was asked is doing UWorld for the second time an over prediction of whether you are ready or not? Okay. So basically, the USMLE Step 1 exam is actually a complete replication of the UWorld. Okay, so the more UWorld you do, the easier it will be for your USMLE Step 1 exam, regardless. Okay, so even if you can memorize, which is absolutely impossible for the first pass of UWorld, which you do, and in your second pass of UWorld, if your scores are above 90%, it does not matter if you remember the question or you do not remember the question, because most of the time I know a lot of students who will do the first pass very well, but in the second pass of UWorld, they cannot keep their scores more than 90% every time in all the blocks. If you can keep your scores more than 90% above in every block in all the scores, then you are more than ready to enter the dedicated period. That's what I meant. But the thing is, I know a lot of students, they think that doing the questions again is not worth it, or it could be a form of cheating, or the fact that you have already seen this question before, you are seeing this question again. But that's not the thing. The thing is, The thing is, the questions in UWorld are more or less the exact type of questions you will receive in your step 1 exam, just a bit here and there, which will be changed out. So the more you do the questions, the more you can understand whether you are ready to take the exams or not. If in the second pass of UWorld, your scores are still not above 75% or more than 90%, you are not ready. If your UWorld scores are more than 90% on your second or third pass, then you are ready to enter the dedicated period. That's what I meant. And when you enter the dedicated period, you're more than welcome to try out any other question banks. To be honest, when I entered my dedicated period, I did MBOS 2. What I would do is I would do 2 blocks of UWorld, 1 block of MBOS, but every day I would do 3 questions, three blocks. That's what I meant. But in order to enter the dedicated period, your second pass of UWorld should be at least more than 90%. That's what I said. Okay. Any more question?
Thank you.
Dr. Jordan, does that answer your question?
Yes, yes. Thank
Okay,
you so
perfect.
much.
You're welcome. Any more question? Dr. Hossam, okay. During the self-assessment exam, if we want to imitate the real exam situation, can we eat chocolate or drink something during the exam? Yes. So what you can do is during the exam, you can take your own food. You can actually take whatever you want to take, no problem. And when you take the breaks, you can go out and you can drink the exam. I mean, you can drink in between the exams and eat whatever you want to eat in between the exams. I personally would take protein bars and dark coffee. And I would not drink a lot of coffee because I would not want to be burned out. I would just have a sip of dark coffee in between each and every break. And I would have a lot of protein bars and nuts and all the types of food, which would give me a constant source of energy instead of a sudden burst of energy, like having white carbs or any types of complex carbohydrates or proteins. What I would do is I would try to stick to simpler diets. For example, throughout that whole 8 hours, I remember having protein bars and nuts and dried fruits and everything which would give me a sustained source of energy for that long 8 hours. So that's that. During your NBME exam, which is a 5-hour exam, you can also do the same thing. I personally did the same thing too. Okay, I would make sure that when I would enter my room or my library where I would study, I would not leave in the next 5 to 5.5 hours. So I would have everything ready. So that's that. Okay. Any more question?
I have 2 questions.
Okay.
When you were doing your UWorld first pass,
Yeah.
you said you took more time to, like, I guess, read the questions. Did you were you annotating more in your first aid?
Was I adding more into my first aid? Is that what
Yeah,
you're
like annotating in it.
like adding extra informations?
Yeah.
Okay. So what I would do is instead of adding the informations in first aid, I made my own notes separately. To be honest, some of the notes which I share with you guys right now are some of the notes which I made when I was preparing. The notes which I share with you guys are some of the notes which I made during my preparation for step 1. And nowadays I have students who are adding it for me. So that's that. But that's what I did. And yes, during the first pass of UWorld, I would actually have my first aid beside me. Okay, since I would do the first pass of UWorld in a tutor mode, I would take a lot of time for each and every question. And if I felt like I had to add some informations to the first aid, I would have added it. But there were times when I felt like I need to write down the entire question because I have to go through the entire question once again. So I would actually maintain a UWorld book. But I would make sure to read that book. Or the same thing I keep on telling you guys is that the UWorld notes, which I have provided for you guys, I need you guys to read that note every day before at least you enter the dedicated period, because in those time span of dedicated period study, you have such little time in your hand in order to do another resource. So you don't want to make that a burden. So that's why I asked you guys to do the UWorld notes right now, because in the dedicated period, I would not do that. The only other time, to be really honest, I'm so glad that you asked this question, by the way, thank you so much. The only other time I would actually read the UWorld notes is before my NBME exams. Let's say if I had an NBME exams on Saturday, I would read the UWorld notes, which I have made on Friday, and that would give me an overall review of all the mistakes I have made. Especially, I would write down the mistakes in the UWorld note copy, and I would try to make sure that I do well in my NBME based on the UWorld notes I have plotted throughout the whole week. Okay, so hope that answers your first question. What is your second question?
Like the NBME, like what order should you take the NBMEs? Like I know I read on like Reddit and stuff that people said to save like 18 and 24 towards the end, and UWSA2 is more predictive. But would you just say like do it in any order, or like is there a specific order?
Okay, it's a very good question. So basically, I have not talked about the NBME. First of all, I just talked about the routine. Okay, now let me talk about the NBME since Dr. Tareen has already mentioned it. NBME, first of all, NBME is something which I hated so much during my preparations. I have no idea why I hated NBME so much, because it was, I mean, I always found NBME extremely difficult. And I know you guys can vouch for the fact that during our discussion, even I mentioned that NBME is something which I find extremely difficult, because I got so pissed off during my actual exam, because I thought the actual exam would be as tough as NBME, but turned out that the actual exam is not actually, at least for me, it was not tougher than NBME. As a matter of fact, it was way easier. So I know that there are a lot of discussions going on regarding whether you should do the NBME, which one should you do before which one, and this and that. But the only best advice I can give you is in the recent NBMEs, the recent ones, NBME 22, 24, these are the ones which are very recent and they have a more higher predictive scores. But then you have a lot more difficult NBMEs, like NBME 18s. They're more difficult, 19s more difficult. So I would suggest that in the last weeks of your preparation, every week, I would advise you guys in your dedicated period to sit for one NBME exam. In the last 4 to 5 weeks, I would advise you guys to sit for the NBME exams, which are more recent. So it does not matter which NBMEs you are doing right now. I would advise you to save the last NBMEs. I mean, the new NBME, the NBME 22, 24, and especially UWorld Self-Assessment 2. I would advise you guys to take UWorld Self-Assessment 1 at the beginning of your dedicated period, because you will get a very good score over there, because it has a tendency of over-predicting your score. I remember my UWorld Self-Assessment 1 score was somewhere around 250s or something like that, and I knew that it wasn't the actual score, because although I did get really happy, but I did know that there's no way possible that I'm getting 250 at the beginning of my dedicated period. But what that did was it gave me a boost of confidence. So you want to enter your dedicated period with a boost of confidence. So I would suggest you guys to do your UWorld Self-Assessment 1 in the first week of your dedicated period, and then let's say on the second week you can do the more recent ones, NBME 24, 22, and on the third week I would advise you guys, or I mean on the fourth week, not the third week, the fourth week I would advise you guys to do UWorld Self-Assessment 2. UWorld Self-Assessment 2 is more predictive of what your score is to the actual exam. So if you're getting 230, 235 in your actual UWorld Self-Assessment 2, then high possibility that if it's a 235, then your score lies somewhere between 240 to 230. And UWorld Self-Assessment 2 is very, very diagnostic of your score, your actual score. So even after that, if you want to increase your score, you can work a bit more harder for the next 3 weeks to make sure that your score gets better, or you can actually calm down if you reach a good score. Let's say you score somewhere around 240, 245 on UWorld Self-Assessment 2 on the fourth week, then for the next 2 weeks, you can actually have a calmer preparation for your exam. So that's that. So that's the best advice I can give you. Has that answered your question, Dr. Tari?
Yes.
Okay, good. Any more questions from anyone else? Any more questions from anyone else? Okay. If there are no questions, then I would ask you to judge yourself whether you are ready to enter the dedicated period or not. How would you judge yourself? Have you guys understood how I want you guys to judge yourself, whether you guys are ready to enter the dedicated period or not? Yes or no? Okay, because you guys will be your best. Just having said that, Let's see who are the students over here. We have Dr. Hussam, Dr. Mudula, Dr. Sabira, Dr. Jordan, Dr. Tareen, Dr. Naud, Dr. Hussein, Dr. Iman. Okay. Okay. So each and every one of you guys, first and foremost, I am really proud of my students on Zoom, and I'm also proud of the students who are not here as of right now, who are listening to this if on the recording. I'm also proud of you guys. So that the thing is, I know that each and every one of you are capable of doing very, very well. Okay, so if you are completely ready, then I would be exactly by your side, not my team, not my team. I will be there personally, trying to guide you in your steps. Obviously, if you're done with the first aid with me and you need help during the dedicated period, that portion of my help to you guys will be proper guidance, and that would be completely free. For example, I would not charge you guys any money for helping you guys out in your dedicated period, and that's the thing which I have mentioned from the beginning. I'm not sure if you guys remember this or not. I told you guys that I would be there for you for every step of the way, but I would make sure that the transactions are clear only for the first aid discussions, and after we are done with first aid, I'm going to be there by you without any transactions or whatsoever, whether you guys need help regardless. So that's what I would do. So I know that each and every one of you are capable of good scores. But with that being said, you have to judge yourself whether you are ready to enter the dedicated period or not. Because once you enter the dedicated period, you cannot do the lectures with us anymore. You have to keep that in mind, because you guys cannot spend 4 to 5 hours of your day doing lectures with 30 more students in a slow pattern, right? Because that's how we do our lectures. Our lectures are not meant for students in the dedicated period. Dedicated period is much more hardcore, right? Over there, you have to focus more on what you need, not on how the pace of the lecture is. So first and foremost, if you have to enter the dedicated period, you have to make sure that you say goodbye to our lectures. Okay, so that's that. But even if you do, please know that I'm still going to be there trying to help you guys out as much as I can. Okay, but also keep that in mind, that if you enter the dedicated period, that you guys cannot do the lectures with us, because if you guys do 5 hours of lectures with us, then you have to do another 5 hours, and then you would be studying for 16 hours a day. That's just too much. So that's that. So let me know if you guys are willing to enter the dedicated period very soon, or are you guys willing to wait till we are done with the first aid, because we don't have a lot of first aid left, at least for the first batch of students. So that's that, and that's all on my discussion for the exam. Okay, it's 2:13 p.m. as of right now. Thank you so much for being with me for so long and putting your time and attention and trust in our lectures and guidance. And okay, so the reason why I am asking you guys to wait is because if you feel like you're not ready, please try to keep in mind that USMLE is a one-go exam. You cannot give the exam again. Okay, so that's that. So if you think you have time in your hand, I would suggest you guys to take some time. Okay. Okay, that sounds good. Okay, so always keep in mind that I'm here. I'll try to help you guys as much as I can, or obviously as long as I live and stuff, right? So that's that. And hopefully you guys will know exactly what you guys need to do, and just let me know. Okay, let me know if you are willing to enter the dedicated period, and I'll help you accordingly. Okay, no problem. I need motivations. Okay. Don't worry, Dr. Wassam, you'll do really well. Okay. You'll do really well along with every one of you guys. You guys will do really, really well. I'm really counting on you guys. Really counting on you guys. Okay. All right. With that being said, you guys are welcome to leave, and that's what I would do too. And I will see you guys tomorrow. If you guys have any more questions, please send us an email. If you guys like our lectures and guidance, please leave us a review on our Facebook page, or let your friends know if they need help, then they can come and join our lectures. And thank you so much. I'll see you guys tomorrow at 9 a.m. Okay, bye-bye now.